Ailshire. There's going to be a lot of epigenetics today. Starting with Lauren Schmitz.
Can you guys hear me? Is this on?
I don't think it's on.
But I can also hear you.
Can you hear me now?
Yeah.
Well, thank you so much for having me and for organizing this. Everyone who helped organize this conference, Jennifer. It's really great to be here. I'm really excited for today to talk to you about epigenetics and also hear what others are working on. I am an associate professor of public affairs at the University of Wisconsin-Madison. I'm a health economist by training, so I'm on the social science side of things. But I have additional graduate training in human genetics. So my work really brings the two together to look at social disparities in health. Okay. So let's start with, why even use DNA methylation to study the social determinants of health? Well, for one, we know it's a mechanism for stable, and that's an important word, stable changes in gene expression that are influenced by the environment. And it's also more proximal biologically to social exposures, which might allow us to see changes in DNA methylation before outcomes manifest phenotypically. So, we can think about earlier intervention, which is really attractive, from a policy perspective. I really like this figure, because we've been talking a lot about these different omics layers. And I really like how this shows kind of how, something is working from the gene space all the way up to a clinical trait. And I put the epigenome in there, kind of in the middle there, kind of this regulatory element space. So, I think this is showing how this is, more proximal to these exposures. And also that, DNA methylation effect sizes are relatively large, perhaps because they're more proximal. Certainly more so compared to SNP effect sizes, right? Like that, vastly different. And so because of this, these biomarkers can be trained in smaller samples, which is a really attractive feature for social scientists because we tend to use, these highly phenotyped cohorts and also cohorts with really rich, sociodemographic information. And so, for these different reasons, it's increasingly being used to construct, as we know, biomarkers of aging and health, like the clocks, proxies of plasma biomarkers, like IL-6 and CRP, and also exposure biomarkers like prenatal smoking and lead. And I want to talk a little bit more about the promises of these epigenetic biomarker proxies. So, this is like in terms of a plasma-based biomarker. This is a quote from this recent paper by Steve Horvath et al. That was published in Geroscience. And they say, another potential benefit of using DNA methylation-based biomarkers instead of plasma biomarkers is that the DNA methylation-based biomarkers are representing a longer average estimate of the biomarker concentration and are not as affected by day-to-day variations that could bias results. So the claim here is that, these epigenetic biomarker proxies may provide more stable signatures than plasma biomarkers. And, as a social scientist, this really captures my imagination because it's going to make it easier to potentially link this to social exposures across the life course instead of a point in time estimate, which might, be picking up more on, day-to-day bounding differences in diet and so forth. And, we see that these scientific claims are already being pitched to consumers. So how many of you are familiar with True Diagnostic? So this is an epigenetic for-profit testing company. It's like the 23andMe for epigenetics. This is a a recent screenshot from their website. So you can see that for about $1,000, you can get all the different aging speed scores.
They had a Valentine's sale last week. Had 2 for 1 sale.
Wow. And they're very excited about this, I know, because I've been talking to a little bit about kind of some of these biomarkers, that you can now get 185 plus epigenetic biomarker proxies. So these are all epigenetic DNA methylation proxies of these plasma-based biomarkers. And this is from their website. They say, true diagnostic measures biomarkers by analyzing their gene's expression. These epigenetic biomarkers are more accurate than similar blood biomarkers because they measure the direct effect on your DNA. And the claim is that these are then more predictive of disease than classical biomarkers. So I'm really interested to, hear more. Well, I kind of put this up to as just a conversation starter too. I think this is fascinating. And so what we wanted to do in this study that I'm going to be presenting is we wanted to see, do some of these claims port over to epigenetic proxies of social exposures. Are epigenetic proxies of social exposures more highly associated with disease than measured or self-reported socioeconomic status? And so this is a collaboration with Jennifer Smith's lab. I really want to acknowledge her and her lab. Also Wei Zhao, who's in the room, and Jessica Fowl. And so this is on medRxiv. We're hoping to hear back about an acceptance any day now. We just submitted the R&R. But if you're interested in looking at a version, there's an earlier version on medRxiv. So just to give you an overview of what we're going to do in this study, first we're going to construct and validate DNA methylation-based biomarkers of childhood and adult socioeconomic status. I'm going to call these C-SES bio and A-SES bio. And then we're going to examine whether these biomarkers are associated with downstream morbidity and mortality, both before and after accounting for both measured socioeconomic status and also the epigenetic aging clocks. So we're really interested to see how correlated these are with the clocks, if they're kind of capturing some of the social gradient that's in the clocks. And then we're going to functionally characterize the individual CPG sites that comprise the biomarkers to assess biological mechanisms and pathways. So kind of using this as a discovery biomarker, right? To kind of look under the hood a little bit more. We know from decades of literature, from many people in this room, that health outcomes are patterned by socioeconomic stressors across the life course, including both individual level social factors and neighborhood characteristics. And this relationship, we think, is likely mediated by health behaviors, exposure to environmental toxicants, and biological changes associated with stress and inflammation. However, I would say the specific mechanistic pathways underlying the biological embedding of socioeconomic disadvantage and its consequences on morbidity from age-related chronic diseases and on mortality are not fully understood. So we're kind of contributing to this. It's very long and rich literature. This is just like a really quick slide on prior research on DNA methylation signature and socioeconomic status. The vast majority of research at the CPG level has been dominated by EWAS, which is no surprise. That's a great way to look at biological associations. There's two really great review papers on these if you're interested, and I have the citations below. I would really recommend those. Overall, I would say research has primarily focused on one domain of socioeconomic status, so for example, education. And so what we do in this study is we're going to use both EWAS and ElasticNet to create these socioeconomic status biomarkers or surrogates or proxies or whatever you want to call them. In particular, the ElasticNet is going to take some of the intercorrelation of these CPG sites into account, which is helpful with prediction. And then we're going to exploit summary indices of SES that measure SES at both the individual and sociological levels. And we're going to do it for both childhood socioeconomic status and for adult socioeconomic status. So to give you a quick data overview, our training data come from the health and retirement study. We have about 3,500 people that we use in that training sample. DNA methylation data was collected from whole blood in 2016 using the EPIC array. Mean age around blood collection is about 69.8. And then our testing data are going to be the multi-ethnic study of atherosclerosis, which, if you're not familiar with it, is another study on older adults in the United States. It was like one of the only other studies out there with DNA methylation data from the US. So I think for this, it's a really useful and important replication cohort. We had about 1,100 people. Importantly, here DNA methylation data was collected from monocytes. So we have a different tissue type, and that was in 2012. And it was collected on the 450K array. And so what we're going to do is we're going to be creating both EPIC-based biomarkers And then we're gonna look at the CPG sites that overlap between the 450 and epic arrays to replicate this in MESA. And you see a very similar age around blood collection. So this is all in older adults. What was also very important to us is that both of these samples are diverse in terms of race, ethnicity, and socioeconomic status. Here's a quick snapshot of our analytic pipeline. It's a little hard to see, but basically first we're going to create summary measures of socioeconomic status. So we're going to create an index of adult socioeconomic status and an index of childhood socioeconomic status. We do independent EWAS of each of these components to kind of see, to compare across the EWAS, but that's kind of a side analysis I'm not going to talk about too much today. Once we create these measured indices, then we're going to do epigenome-wide association studies, or EWAS, of these SES indices, one for adult socioeconomic status, one for childhood. And we're using that kind of as a pre-selection way of choosing CPG sites that are, correlated in FDR, p-adjusted value less than 0.05. And importantly, I think that we know are associated with the biology. Because as I was talking about last night with Colter, if you just do elastic net, you don't know what it's choosing. It might not be biologically informative. And so I think this first kind of EWAS step to pre-select some CPG sites is an important one. And then from there, we're going to put those CPG sites into the elastic net procedure to select independent CPG sites that are associated with adult and childhood socioeconomic status. Then we're going to generate the biomarkers. We test these in MESA. And then we're also going to look at longitudinal association with health and mortality. So in HRS, we're going to be looking at health outcomes 2 years after blood collection. In MESA, I believe it's about 6 years after blood collection. In terms of how we construct the socioeconomic status indices, for adult SES, we're going to be taking the average quintile of non-missing education years, household income, wealth, occupational prestige. So here we're bringing in measures from the General Social Survey that were matched to 3-digit occupation codes. Neighborhood socioeconomic disadvantage. These are these amazing measures from Jennifer on the contextual environment in HRS that we use, including a summary of 6 census tract level neighborhood metrics, including that reflect kind of education in that census tract, occupation, income, wealth, poverty, employment, and housing. And then we also look at the neighborhood social environment. So these are self-reports of aesthetic quality, safety, and social cohesion, again, that are aggregated to the census tract level. So we have both, self-reported measures and also these more environment kind of local-based measures, sociological measures. All these variables were averaged over HRS waves up to blood collection, except of course education. For childhood SES, we're much more limited in the data that we have. So we have parental education. We took the average of non-missing parental education, which we coded between 0 and 4 based on these different intervals. And then we also have this childhood financial strength index in the HRS that we use. So whether when you were young, your family ever moved, received financial help from relatives, whether the father had a period of unemployment, and self-reported poor or varied financial status in childhood. And in all of these, a higher index was coded to be greater disadvantage. So higher index is worse. And I will say too, we also did like PC-based versions of these, and we got very similar results. So here are results from the EWAS of adult SES Manhattan plot. And for the EWAS model, you can see controlled for age, sex, race, smoking. Smoking was like we were constantly controlling for smoking, right? Because we really want to make sure we're not just picking up smoking. White blood cell count proportions. We did include genetic PCs in there, and also technical covariates. And then once for the ENET models, then we also pre-adjusted the CPG sites for all those covariates that were in the EWAS model. And then we use those selected sites to predict adult socioeconomic status. What you see here that's interesting is that the sites in the EWAS that were over 0.05 p-value, these are also ENET sites that were highly selected by ENET. So for most part, the sites that were in the top ranked at the top for the EWAS were also sites that were picked by ElasticNet. So what we see for the adult SES biomarker It chose 25 CPG sites from over 69,000 CPG sites with P less than 0.05 and the EWAS. In the HRS, the cross-validated R-squared was around 5.6%. In our replication sample in MESA, it predicts 4% of the ASCS variation, which doesn't sound like a lot. But, I used to work a lot with polygenic scores and, we needed 1 million people to get to, something decent. So this is, this is in 3,500. The childhood SCS biomarker was not as predictive. So I think in part, we have a lot more measurement error with this score. We don't have as rich of information. But ElasticNet did choose 17 different CPG sites than what was in the adult biomarker from over 88,000 CPG sites. It had a cross-validated R-squared and an HRS of 3.4%. In MESA, unfortunately, we don't have those financial components. So in MESA, we can only see how it predicts with parental education, and that was, around 0.5%. I want to show you a quick correlation table. These are correlations in the HRS. They were very similar in MESA. A few things I want to point out here. First, with the blue correlation coefficient, that's the correlation between the two biomarkers for adult and child socioeconomic status. And you can see they're not super highly correlated. So they do seem to be capturing some different information. Down there with the orange, what I want to show you is how these are correlating with grim age and Dunedin pace. And what's really interesting here is that you see much higher correlations with the adult socioeconomic status biomarker than you do with associated with just measured SES. So these do seem to be capturing something about a social gradient in these clocks. And I think what's also really interesting is that we see a much higher correlation with Dunedin pace. And that was even higher in MESA. I think it was like 0.63 in MESA for that Dunedin pace. So maybe Dunedin pace is capturing more of this like social gradient, which could make sense. It's across several time points in the life course, the way they trained it, I'm sure. And then you can see with the green correlation coefficients, we just don't see that with the childhood measures. So there, actually measured childhood socioeconomic status is more correlated with the clocks than the biomarker. This figure, sorry, there's a lot going on here, but what this is showing is these associations with downstream morbidity and mortality. So we looked at 5 different outcomes: number of chronic conditions, cardiometabolic conditions index, self-reported health status, mortality, and also dementia. Model 1 is showing coefficients that control for age, sex, race, smoking, and ApoE4 carrier status. That was for dementia models only. And then model 2, we added in measured SES. So we wanted to see, is this still predictive after we control for what we already know about these people, right? And we see that, it does seem to be capturing some unique information for the most part. We don't see a lot of attenuation in the coefficients after adjusting for measured SES. What you see here too is that they're predictive across the different types of platforms. So we have the HRS Epic version, the HRS 450 version, and then the MESA 450 version. For childhood socioeconomic status, those did not replicate well at all in MESA. So we don't really see much in MESA. And then interestingly enough, again, or maybe, based on what we already kind of know about social effects on health, we see more in terms of chronic conditions, cardiometabolic conditions, mortality. We don't see much for dementia. And then just to note that further adjustment for epigenetic aging measures, so then we tried adjusting for grim age, Dunedin pace, also did not fully attenuate these associations. So this is capturing unique information that's not already in the clocks or in measured SES. This is just a little regression table showing another thing that I thought was interesting, which is that in MESA, our replication sample, we see that the adult socioeconomic status biomarker is actually a better predictor of cardiometabolic conditions and mortality than measured ASCS. Which isn't the case for self-reported health status or dementia. So again, we do feel like we're picking up on these, social signatures of health that have been reported a lot in the literature in terms of chronic conditions and mortality. We then went ahead and did some functional characterization of biomarker CPGs. We have a lot of fun figures and everything in the paper. I didn't want to kind of overload that on the slide, so this is just a rough summary of all of that analysis. But if you want to look in the paper, we have all the genes and everything. So, but this is just going to be an overview. So we see that these CPG sites are enriched for genomic features that regulate gene expression. So DNase hypersensitivity sites, enhancers, promoter regions, and EQTMs. And so what this is telling us is that these are playing a functional role. They are doing something, that's impacting gene expression. It's also associated with gene expression in pathways related to chemotaxis, T cell activation, immune system response, cell migration, and cell death. CPGs from both biomarkers were associated with prior EWAS of age-related chronic diseases and inflammation. But we also see some distinct signatures between the two, and I think this is really interesting. So for CSCS bio CPGs, these were associated more with immune system dysregulation and with autoimmune disorders. And for the adult biomarker, we see more associations with inflammation, metabolic disorders, cancer, and health behaviors. So this is kind of supporting prior research that has shown childhood disadvantage as a risk factor for immune dysregulation and changes in T cell activity, which in turn can increase the risk of mental and physical health issues in adulthood, including cardiovascular disease and cancer and so forth. So I thought that was interesting based on what we know from the literature. So now I'm going to wrap up with a few slides and kind of just put some discussion points out there, things to talk with you all. So just to wrap up, we developed and validated DNA methylation biomarkers for adult and childhood SES. The ASES bio explained around 4% of the variance in adult socioeconomic status in a replication sample. ASCS bio, both in HRS and MESA, and CSCS bio and HRS were associated with chronic disease, cardiometabolic conditions, and mortality, even after controlling for measured SES. And then, despite the modest variance explained, we think these findings are significant due to the fact that we were able to replicate across tissues and platforms. So that kind of demonstrates robustness. And also the ASCS bio outperformed ASCS in predicting cardiometabolic conditions and mortality. So that's suggesting it might have some, clinical utility, or, it's telling us something new. And then finally, it provides insights into biological mechanisms and pathways linking socioeconomic status to health. So CPG sites are associated with dysregulated immune system responses, inflammatory pathways, poor metabolic functioning, chronic diseases, and cancer. And then I want to talk a little bit more about, the epigenetic aging measures and their overlap. So we know epigenetic aging measures may reflect social gradients in health by capturing the cumulative impact of social and environmental exposures on biological aging processes. And we did see that the SES biomarkers were more correlated with grim aging and median pace than measured SES. And controlling for epigenetic age did not fully attenuate the SES biomarker associations with health. So we think these are capturing additional information. Maybe this is a little bit too much information, but this is showing how, depending on the biomarker and the health outcome and which score we were controlling for, we saw attenuation of anywhere from 1% up to 61%. So this also does need to depend on the outcome that you're looking at. But what do we take away for this? Well, in some studies, controlling for SES biomarkers may reduce bias from SES-related confounding in these epigenetic aging signatures. So that could be another, application of these. In terms of limitations and challenges, I think larger, more, we already have this great representative sample in the HRS, of course. I was like, representative samples are going to help improve prediction and replication. Especially those that are phenotyped well, for childhood socioeconomic status and adult socioeconomic status. We see that these SES measures used to train biomarkers, they could be subject to recall bias and measurement error. So, better kind of maybe objective measures of SES in addition to self-reported measures would always be helpful. Causality. So I really want to, point out that these are correlates of socioeconomic status. They're not causal. And some of these CPGs may be uninformative. Generalizability may also be limited, but I think this is both an opportunity and a challenge. So it'd be really interesting, I'm hypothesizing that these are specific to the US context, but it would be really interesting to see if these validated in other countries, if they replicated in other countries, I should say, that have very different social welfare systems, policies, and programs. And environmental contexts, right? it's also trained in older adults, so it may not generalize to younger populations. Technical factors, I know Colter's gonna be talking a lot about this, you know, batch effects across labs, all this kind of stuff. Unreliable probes, array differences, this is also a challenge for generalizability. And finally, tissue specificity or replication in other cell types would also be interesting. And then, you know, to really get at biological independence, I feel like we need to do gene expression analysis to confirm these distinct childhood adult SES biomarker pathways. I think that would be an interesting next step. So yeah, just to outline a few next steps, you know, examining associations between SES biomarkers, CPGs, and gene expression. Evaluating SES biomarkers in each racial or ethnic group separately. So I think, you know, we just pulled everyone together, but I really think, especially when you're talking about socioeconomic status and the environmental context, we could see different patterns by race because of, you know, social disadvantage that are, you know, experienced differently across race and ethnic groups. And, you know, from a developmental life course standpoint, you know, investigating the developmental trajectory of these methylation signatures across the life span, so seeing how these evolve across children and adults. And then, I think it'd be really interesting to see if these generalize to other countries. So methylation signatures may differ across contexts due to distinct policy environments. And so thank you, that's all I have. And
thank you. I haven't kept up with the epigenetic literature in the last 10 years, and a lot has changed how we talk about race and how it interacts with SES. And you hinted at that a little bit to look at race separately. I'm just curious why you add race or you control for race in your model at all. Is there any biological reason to do that, or
I think there it's more the social environment, because I think race is a social construct.
Exactly.
Yeah.
And so if you don't include it in your model, you would actually see a larger effect of SES, right?
That's true.
I'm
Yeah.
curious what the choices behind including.
I mean, I think we were worried about confounding, you know, in that sense by race. I think, but you're right that it does take up some of the you know, it does eat up a lot of that predictability. So yeah, I don't know. I guess that's a question. I think for us it was important for reasons of confounding, but
I think it would be great to look independent, also in between groups, but you're already doing that by controlling it alone.
yeah, that's true. But yeah, you're right that it would be interesting to see what we get when we don't control for race. Yeah.
I have a very similar question. So I'm still trying to figure out when to control for things and when.
Yeah, me too. Yeah,
So my concern was with smoking and cell type control. You controlled those out before the training step, right?
Mm-hmm. Yeah, because of this huge social gradient in smoking, right?
Yeah,
and
but
how
is that not the pathway that you're interested in?
But we didn't want to just capture, because these smoking CPGs are so,
Right.
you know, their effect sizes are so large.
Yeah.
We didn't want it to just be overwhelmed by smoking. So we wanted to see, are we capturing stuff in addition to smoking?
Okay.
You know, I think those are also, you know, so
Right,
you
yeah,
could
yeah.
also classify those as being social, right?
Yeah.
But I think we wanted to see, because there's already been so much literature out there,
Mhm.
are these in addition, you know, do we see additional pathways that aren't just related to smoking?
Yeah, because my instinct is always to then train and then control smoking out in the next step, but I see what you're saying.
Well,
I'm trying to-
we also control for the
Sure, yeah, yeah.
regressions and yeah.
Yeah, I mean, I don't feel like I found a satisfactory answer at all, so I'm very interested,
Yeah,
what
I
other
think,
people are thinking.
yeah, and Jennifer Smith also, you know, felt
Yeah.
strongly, we talked a lot about that, and she's like, no, we got it in the beginning because they're just, the effect sizes are so huge for
Sure.
smoking, it's, yeah.
Yeah.
But in a sense, if you're trying to do for a population
Mhm.
and what is the overall effect of SES, you kind of want to leave it in.
Yeah,
I mean, I understand epidemiology, you want to get everything out before you look at what you want to look at, but I do think people who do populations want to
Yeah,
get like the complete picture first.
also
I mean,
wonder,
you could, yeah, it'd be interesting to do it both ways and compare and see, yeah, I think, yeah, again, we were just kind of concerned that we were just gonna be capturing smoking signatures and.
Well, but if
Something
that's
additional.
the story.
curious about what like, the pure effect of SES would be. That's because I think of smoking as an SES.
Yeah, no, it is
pathway.
on the pathway, yeah,
Same with like cell type,
yeah.
that could be things like exposure to pathogens.
Pathogens, yeah.
Yeah, cool.
So I love this and harkens back to things that we talked about when you were.
I know, yeah, he was one of my postdoc mentors, so this is, yes, I did.
So this is great. Kind of a broader question though is to think, so you have SES at different time points in theory. HRS doesn't have the greatest childhood measures of SES. But the thing I struggle with sometimes is like, yeah, so let's do child cohorts and do SES in childhood and create those measures, right? Imagine someone doing that. But is that really the right thing? Like a lot of times we think, oh, let's get the exposure closest, the methylation time closest to whatever exposure we want, because that'll be the biggest signal. But then I think, but actually, don't you want the one that is like the most persistent?
Yeah,
Because a lot of these signals fade over time. I don't know.
No, and I think that is, I'm always thinking of these things too, back and forth of, what. So yeah, I think there was a lot of measurement error too in this,
There's
in
a
the
lot of
childhood.
this one, yes.
So it's, I feel like we couldn't really evaluate the effect of childhood socioeconomic status, and that's where, maybe going into the childhood cohorts, you would get a little bit richer stuff there, but.
Or the British
Yeah.
cohorts that have
Yeah.
better childhood
Yes,
data, but later life.
Right,
the-
yeah,
that would
doing
be-
it tested a few different ways that way,
Yeah.
because we have childhood measures of SES too, or poverty, but.
Yeah.
But I don't know, it's just a thing I think about all the time, because often we say like, let's get the time point closest to the exposure.
Yeah.
But the reality is a lot of that might fade away because it's really not important. It's like a temporary signal.
Yeah, no, I think that's a really good point. I think if folks know other cohorts that they want to try to replicate it, or, I'm a very collaborative person, so yeah. I'm not-
Very interesting talk. I wonder if you.
Alex and then.
Yeah, sorry.
Yeah, go ahead, Alex.
Yeah, thank you. Very interesting. I wonder if you checked how well your score is correlated with aging, with just chronological age. You saw that the variation with SES is like 4%.
Yeah.
So maybe your score actually captures mechanisms of aging and health,
Mhm.
and they in turn predict SES because we know that health and SES are highly correlated. I wonder if you checked that, and that would help.
We just control for age and the EWAS and then, the controlled out for age in the CPG sites before we put them into ENet and then in all the regressions. So hopefully it's taking out some of that, but yeah, it would be interesting to look at the correlations.
I mean, since your score is highly correlated with Indian pace,
Yeah.
I'm sure there is some correlation with age, even though you adjusted. So I wonder,
Yeah, that would be interesting to look at.
I guess it's similar to the earlier discussion about what do you adjust for.
Yeah.
I mean, if you adjust for smoking, ultra processed foods, I mean, from an exposome perspective, it's a really interesting study. But then what are you actually interested in? Is it health you're interested in? Because it seems you're picking up health status, but or are you interested in the environment? So maybe that's what you need to.
So thinking more about, so say more, so not,
So maybe you should be adjusting for health.
Well, start with you, I'll start to eventually pull every.
Yeah, no, I mean, discussion, no, thank you for these comments and, thank you.
Thank you very much.
So good morning, everyone, and I just want to say thank you to the organisers for the kind invitation to come and join you here today to have a discuss this. It's been really exciting. Lauren provides a nice segue for me into my presentation here. We're going to try and decompose the social gradient we see in these next generation epigenetic plots. So just to provide a bit of context, we know that individuals from more disadvantaged backgrounds get diseases earlier and they die earlier on average compared to their more advantaged counterparts. They also live a larger proportion of their shorter span of years in disability and in ill health. So we kind of know that biological time at least seems to run faster for these lower SES groups. And with social epidemiologists in the audience, for years we've looked at the pathways here through which this operates. So neo-materialistic factors like the quality of your diet or the quality of the household you live in, lifestyle factors like smoking and so on, but also things like psychosocial stressors, adverse childhood events, occupational hazards and so on, kind of broader exposome, I guess. And then kind of luminaries like Teresa, like Eileen, like Steve Cole, have been looking at these mechanisms through which the social environment gets biologically embedded into the tissues and the cells and the molecular biology. So I've done some work before on allostatic load, others have worked with telomeres, epigenome, transcriptome, proteome, and so on, as we saw yesterday. And as Lawrence has outlined, one of the best candidates that we have currently is DNAm methylation, which kind of determines how well genes get expressed or repressed like that. Now, we've heard about clocks a little bit just in here a lot today, as Eileen said. So, and just a quick revisit of them, what we're doing with the clocks. So the first generation clocks were built to measure the passage of chronological time, correlated very highly with the calendar age. And then the second generation clocks, and Morgan Levine is credited here for this, but these are built using kind of clinical biomarkers like phenoage and grain age, and we know that they predict health state better than others. We have the putative third generation clocks, I'll come back to that. This idea that they have characterized change in the clinical biomarkers, but the signals distilled in the DNA methylation as a single time point, and that is important. And then finally, we have the fourth generation clocks, again, the so-called fourth generation clocks. So things like cause age, dam age, and adapt age, which use Mendelian randomization to identify CPG sites that were meant to be causally related to aging. So we haven't done very much work yet profiling social gradients in these next generation clocks. So that's what we're going to do today. So sorry for this expression from I think it was the American Foreign Minister at the time. So the known knowns. So what do we know? There's very well established social gradients in second generation clocks. So here, for example, using 5 different measures in tilde, such as class, education, income, housing value, and neighbourhood deprivation index. And we can see a very clear social gradient across most of these measures. I picked the most advantaged, professional, managerial, tertiary level education, highest income, and so on. And you can see the epigenetic age acceleration residual here for grim age. So I thought this was a fairly well established pattern, so I'm not going to bother writing this paper up. And then I was asked to review this paper earlier this year, disclosure, aging research reviews, and it looked at the relationship of SES with lots of the established clocks, and the patterns weren't as clear as I thought they were in the literature previously. So you can see that they're all pointing to the right here, at least in the context of SES, but there's a lot of variability around that signal too, right? So we then derived the Dunedin-Pace clock and the causal clocks in tilde, getting excited about this. And then this paper pops up, and it's from Eileen, who beat us to the punch. So Eileen had done this already in the HRS cohort, and she looked at the relationship of lots of clocks, not just the causal clocks, with the age acceleration residual. And what you see here is actually there's not that strong a signal in the HRS for cause age, for adapt age, and for dam age, these next generation clocks. So just to provide a bit of context here, dam age is meant to indicate molecular damage, and adapt age is meant to indicate geroprotective mechanisms, and cause age is kind of like a balance of the two. Okay? So in fact, we found here in the HRS that some of the relationships are actually paradoxical. They point in the opposite direction to what we might expect with SES. So what can we do that's different? So we're going to look at this as well in tilde, just to see, do we see similar relationships are different? So it's accelerated aging due amongst more disadvantaged groups due to this accumulation of molecular damage or a failure of that maintenance repair capacity that you see with adapt age. And then I'm going to do something slightly different, maybe to what Eileen has done anyway. Is how modifiable is that epigenetic aging signal across the life course if you're upwardly or downwardly socially mobile? And how stable is that social gradient over time? We now have methylation at time 2 in tilde, so we can actually look to see how much change you see across the social groups over a 4-year span. So this is the kind of broad analytical approach I'm going to take in the paper. So I'm just going to show you what those social relationships look like in the tilde data set. I'm going to perform a mediation analysis to try and unpick or explain away the social gradient. I'm going to examine the impact of social mobility on epigenetic aging using diagonal reference models. And I'm going to look at change. I'm going to do this really quickly. So for those of you not familiar with the tilde cohort, it's a nationally representative cohort study of aging in Ireland. At baseline, we had about 8,500 individuals. So our sampling fraction is 1 in 156 of the Irish population, aged 50 years of age and older. So we're really population representative. And like the HRS, we have a computer-assisted personal interview, a self-completion questionnaire. But we also have a health component where we bring people to a health centre in Dublin, and we do about a 4-hour workup with them, so they do a really detailed phenotyping during that visit. we collect blood, and at each wave we tend to do things a little bit different. At wave 3, we had hair for profiling stress hormones. At wave 6, we collected stools, saliva, fluids, and so on. And we're just rolling into the field now with our wave 7 suite of data collection. Now, till the epigenetic sample is split across time points, we initially did about 500 individuals in Italy as part of a European consortium project. We were getting ready to do our time 2 samples. We thought we had enough EPIC1 array to do everyone, and then we found out Colter had cornered the market. So then all of a sudden we had to like reconfigure our sample. So what we had is of the original 500 and 490 individuals, we repeated them on EPIC2 at time 2. Our new sample, we did all of those on EPIC1 on time 1 and EPIC1 at time 2. So at least everyone at time 1 has been done on EPIC1. We have a repeat sample of 46 individuals on EPIC2 across 3 time points. We have to have data now to 14 years after baseline collection. And then we have a repeat sample too. So this is for the molecular biologists that love this sort of stuff. I'm not going to talk too much about that, but just to give you some indication, we have about 950 samples at time 1 here, which I'm going to present today. Lots of this stuff was done by Rob, you're going to hear from later. So the processing of the tilde DNA samples you're responsible for. We hopefully are going to be working on a few papers looking at the impact of different pipelines and their impacts on the clocks. But what I just want to say here is what we did is we selected sites that were common to EPIC1 and EPIC2 because we had to deal with that change in chip type over time. And the effective age is regressed out. So in all these analyses, I'm going to show you the extrinsic epigenetic age acceleration residual. And for the, I've left out Dunedin pace here, but for the causal clocks and with RIMage, you can see here at the top, yeah, this is the number of CPG sites in each of the clocks when they're originally developed. You can see there's actually very limited overlap across the clocks, not a huge amount of RIMage anyhow. And then when we select sites only common to EPIC1 and EPIC2, we actually start to lose lots of sites across these clocks, right? So you can lose up to 40% of the sites in these clocks. And that's important when we come back to talk about the results. So just to start, this is the the epigenetic clock intercorrelations for the different clocks. So what you can see here straight away is that damage is highly correlated with cause age, as you might expect, but also with RIMage and inversely correlated with adapt age. Remember, it's the geroprotective function. That's what we expect. Not particularly strongly related to Dunedin pace in the tilde cohort. And then we mentioned about looking at the impact of the social gradient. So here what I've done is I've converted everything to standard scores because Dunedin pace is a rate clock rather than expressed in years. So these are standard scores. But what you can see is the social gradient is much steeper here, much more pronounced for RIMage compared with Dunedin pace. What does it look like then for the clocks? Well, this is where we get a difference from the HRS. Now all of a sudden we see that cause age is associated with epigenetic age acceleration. That damage is associated with increased molecular damage, as anticipated. But you also see that compared to the third level of tertiary educated groups, individuals from more lower educational backgrounds tend to have less geroprotection. So that may index a failure of repair capacity very early in life. So if Teresa was here, she'd enjoy this. It's almost like allostatic load, the seesaw again, isn't it? So maybe we can think of aging like that. So, of course, we have an effect of education on clock age. We don't think it's direct. It's being mediated through some process, lifestyle factors, and so on. And what I've done here is I've just broken this up for a number of different risk factors for accelerated aging. So this is your smoking impact years across the different educational groupings, much more prevalent in the more disadvantaged groups, or sorry, the lower educated groups. In fact, for almost all of these risk factors, you can tend to find is that individuals from primary educated backgrounds do worse, and there's a clear social gradient in each of these things, with the exception of alcohol consumption, which is the key hazard. It tends to be more common in more affluent groups in Ireland, or problematic drinking. And just to talk about the inflammation Z score, we have a battery of cytokines, chemokines in tilde, and I selected about 6 or 7, I can't remember which ones they were now, CRP, IL-6, GDF15, and a few others, and put them in the inflammation Z score, and you can see here the individuals from primary backgrounds at higher levels of inflammation. Then what I do is I just do a decomposition analysis. So using KHB mediation. So just to talk you through a little bit of this, because it's kind of rinse and repeat. The reduced model is essentially the total effect, right? So it's telling you that it compared to tertiary level, compared to those with a third level education, those with primary level education are 3.25 years older on the Grimm age clock. The full model then is the effect when you adjust for all of the mediators, okay? You think of that as a direct effect. And then the difference model is a proportion of the effect that's mediated through that set of mediators, okay? So you can see that when we adjust for all of these mediating factors, you explain away 46.3% of the social gradient in Grimm age. And the significant indirect effects are coming through smoking pack years, as you might expect, but also social network size, that's the Berkman sign, and the inflammation factors, so they're all important here. We do a little bit better with the leading pace. Now we explain almost all the gradient, no longer significant. We explain nearly 80% of it. And again, kind of consistent patterns, smoking pack years, waist height ratio. Waist height ratio is different here. It wasn't for the previous block of this year. As a CSD depression score, and inflammation is explained a little bit more in the context of the leading pace. Cause age, we explained about a third of the effect, none of the indirect effects are significant. And then this is where things start to get difficult, right? For damage, none of these kind of classical risk factors seem to explain much of the gradient, so no significant indirect effects. And for adapt age, when we adjust for this constellation of risk factors, we release a suppression effect and the differences get bigger. So a bit counterintuitive, but that's what we found initially. So the next thing, I don't know how I'm doing for time, is how modifiable is epigenetic aging. So this is a diagram I took from a US paper, believe it or not, back in 1916. It's kind of looking at a counterfactual. The returns to this man for going on in education, the communist dream. So in the top arrow, he goes on and improves his education, and you can see he's aging more gracefully over time. In the bottom example, he doesn't go on in his education, and he's a bit more weathered and grizzled looking as he ages. So we can take that type of scenario and apply it in our data sets. And the way we tend to do this is you tend to use a cross classification of some childhood measure, like your father's social class and then your social class, or your father's education and your education, and then you can look at the age acceleration across these groups, right? And you can make assumptions about whether mobility is actually good for you or not here. So you can see here the stable tertiary, their father had a third level education, they have a third level education, are aging more slowly here than those who got primary level education and their father primary level education. Okay. Now, the difficulty with that is because mobility is the difference between your childhood position and your adulthood position, right? You run into the issue of you have an problem with identification here, right? How do you separate the mobility effect when they're linearly dependent on each other? So one way you can try and get around this is to use diagonal reference models. So the beauty about diagonal reference models is that they use the socially immobile groups as the most suitable point of reference, as they represent the true characteristics of the given class and you stay in that position over time. And then the effect of moving from class I to class J is a way to sum the diagonal effects. And just to explain that a little bit better, so you have this individual here, his father had a tertiary level education, he now has a primary level education. So to make an assumption for him what his epigenetic aging looks like, it's a way to sum these diagonal effects. And we also calculated weights for them, which gave the proportional weight applied to childhood and adulthood, and they sum to 1. Right, and then once you've done that, you have the weights, and you also have the coefficients in the diagonal part of the model. Then you can make assumptions about what the impact of mobility is over time. Just to show you that in Ireland, very few people were downwardly mobile. This is shown in the bottom part of the diagram. You see kind of sticky floors and sticky ceilings. All right, individuals who have advantage tend to keep it across the life course. Those who are more disadvantaged tend to keep that across the life course too. But in Ireland, we had quite a lot of social mobility because we introduced an Education Reform Act in 1967, which made secondary education free for everyone. Before that, you used to have to pay for it. So the direction of mobility tends to be mostly upward in Ireland. So what do we see here? How do we interpret these models? So what you're showing here is the coefficients for epigenetic aging for those who are stable in their class positions over time. And you can see that the stable tertiary is associated with slower aging, stable primary is associated with faster aging, and you can see a clear gradient. Then the coloured code bar at the bottom indicates the proportion that's explained of your epigenetic aging by your father's education and by your own education. And you can see here it's about 25% is explained by your father's education and groom age, and about 75% is explained by your adulthood, your adult education. For PS, it's kind of similar. Again, you see a clear social gradient across those groups. And here, if childhood, if your father's education explains about a third of the effect, again, dominated by your own education. And then we start getting the problems again with the clocks, right? With the causal clocks. Now, all of a sudden, for cause age, 100% is explained by your childhood education, right? Your epigenetic aging on the cause age clock is explained by your childhood. And for damage and for depth age, again, you tend to see both are important, but it's a bit more dominated by childhood relative to adulthood. So a different pattern that we get for the other second generation clocks. And this is essentially the same thing shown in the table. I couldn't think of a nice way to show this. But what this answers the question about mobility. Once we do that, we separate those elements in our table, what we find here is upward social mobility is not associated with slower epigenetic aging. It's not significant in terms of your epigenetic aging. And interestingly, if you're downwardly socially mobile, you seem to be aging slower, right? Paradoxically, on grid age, on damage and adapt age, you have more geroprotection. And remember, epigenetic aging isn't constant over time. The ticking rate is meant to be faster in early life, so this might indicate some level of critical period effect, or just the data is really noisy for those clocks. All right, I am going to wrap up soon. So the change in the epigenetic aging, so we've got 833 individuals for whom we have epigenetic age at time 1 and time 2 until, which is our wave 3 sweep of data collection. There's about a 4-year gap. So the vast majority of people who came along had a 4-year gap between their time 1 assessment and their wave 3 assessment. And you can see that here in the top. And then down here, it just shows you the epigenetic age acceleration for these groups over time, so changing them over time, okay? And you can see by the legend here that some of them are changing quite a lot. So young age, the range is between -20 up to 80, right? For damage, it's -150 up to 50. So some individuals are characterizing massive epigenetic aging over that time period. You tend to find that for groom age, much more closely bunched. Very few people hanging out on the fringes here. And again, when we look at the change in epigenetic aging by education, we see that there's actually very little change over time. And remember, this is a very short time span. We're looking at change. It's only 4 years. In fact, it's only for cause age you see any level of acceleration. So if you look at the cause age clock, you can see that over that time, 4-year time span, the individuals from primary level education relative to tertiary level are aging faster by about 0.85 years, okay? But then when you look at grim age, you can see that the age acceleration is negative, and that suggests that some level of catch-up among the tertiary educated relative to the primary educated, right? So in the first instance of going, okay, we don't see much change, but maybe remember the mean age of the sample is 62 at the baseline. So maybe just so much aging's already occurred. How much do we really expect to see over that 4-year follow window? And as I said, if anything, we see the social gradient more slightly using the better clocks or like room age here. So there could be selection effects in the data. We can back for the health assessment, they're included at wave 3. But the other thing is for those epidemiologists, like there is this idea of age is leveler hypothesis. So if you're in really good health, it doesn't matter. Time's going to catch up with you and something's going to get you in the end, right? We all die. So maybe there is some level of catching up here. So just to finish. So we saw an established social gradient in epigenetic aging across all of the clocks. And we said that this differential aging may be due to both accumulation of molecular damage and loss of maintenance of repair capacity. So I think that's important. I'd like to dig into that a little bit more. So we're hoping to do some of that in Tilda. The classical lifestyle, psychosocial and biological risk factors explain a substantial portion of the gradient in second generation clocks, and I include Mermaid and Dunedin pace as one of those, but not in those causal clocks. For the diagonal reference models, we find that both childhood and adulthood SES is important for epigenetic aging, but adulthood predominates in the context of Dunedin pace and Grim age, and childhood for the causal clocks. So again, you might be picking up different things. And my own view on this is that the causal clocks remain a bit of a black box, and I think there's been enough work done with those clocks. And just to finish, what I tend to find with lots of the work that we're doing now is that the Dunedin pace and Grim age look very similar. I think they're both second generation clocks. I don't think we have really moved the needle on the clocks. I think we're still stuck at the second generation. And if there's a third generation clock, it's the Horvath and tightly used Pan-Lamanian clock, I would suggest. But with that, I'll finish. Thank you.
I was actually very impressed that these causal clocks associate so strongly with social gradients. You know, we've been doing work in airwave looking at smoking on the clocks, and obviously, you know, Dunedin, huge effects, but no effects on the causal clock. So we start to wonder what's going on. But here, this is showing quite convincing that they are picking up things. Do you think actually smoking doesn't actually affect aging?
Oh no, it absolutely does.
We prove that.
I mean, what
Worse money, yeah.
Could be economists if we want to have that as our statement.
I mean, I guess what I'm saying is, you know, the causal clock, the first generation clock, are meant to pick up biological aging processes, whereas smoking causes specific pathology. So is it something sitting on top of aging, and should we be saying, well, actually, we shouldn't be worrying that these clocks that are meant to pick up aging don't associate with smoking? You know, because maybe that's telling us something, that actually smoking isn't making people die because it's accelerating their age, making people die because of pathology. You know, this is a very philosophical question about going round and round and what acknowledging. That was my comment. Maybe it's too
technology there, Oliver.
Yes, exactly.
Maybe for me at this time in the morning.
Maybe it's too big a question.
But what I will say, so the causal clocks, so I find them interesting, right? I like the concept of it's not just acute molecular damage, it's also failure of repair capacity, right? And you can think about things like this. Autophagy would be a good example of that, right? So I don't think we measure that particularly well, not in the population studies. We don't know how to do that. I don't know other people out there.
We've got an attempt now.
Oh, we do. Okay. So that's good. So I think that will be one of the processes that we'll be looking into. My PhD students look at work on this idea of housekeeping genes, you know, cleaning up the debris and detritus and so on. Well, with the causal clocks, so we see the social gradient, but then the causal clocks don't predict the age phenotypes until that. So you get like striking. They don't know. I mean, the Grim age and Dunedin pace, to be fair, both predict the age-related health outcomes really well. The causal clocks don't. So I do wonder what the causal clocks are picking up. Maybe it's some signals from childhood that we just haven't, maybe it's viral exposure. We were thinking about some of the things that we haven't accounted for in the data that might potentially explain that. So that was one of the areas that we do have VIR scan data in Tilda, so we can look at your viral histories. And so we haven't done it yet, but we could look into that. So I don't know if I'm truthful. When I first saw the social gradient, because it wasn't as apparent in the HRS cohort, and we saw it in Tilda, I was like, oh, this is really interesting. We're getting different signals. Maybe it's due to the different racial composition. We have a mostly, you know, European ancestry white cohort. The causal clocks are trained in Generation Scotland. So I was going, maybe that's why we see it, right? But so I don't know. I just think there's lots of work for us to do to unpick those
But
clocks.
shouldn't we publicly say what we've learned in the last few days about the difference in the causal clocks between us? Because we've done we've done loads of clocks. We normally get exactly the same thing in Ireland and HRS. When we found something different in the causal clocks, we started looking into it. And is Rob here?
Yeah.
So Rob and Eric did quite a bit of work. I mean, why don't you at least briefly say what we know at this point?
So I just went in and took 10 random HRS cases
But,
and just looked at.
Start with the idea.
Well, yeah, so one of the issues is just normalization and the DNA methylation.
It's a technical thing.
Yes, so
It's a
we
technical
use noob normalization and you used Illumina. And apparently they use
Use
quantile
quantile.
normalization.
Yeah,
And
three
so,
different approaches to normalization.
Yeah.
Probably most people in the room are never gonna bother to be normalizing the basic data, but this is a source of
so yeah,
differences.
especially for individual samples that had particularly poor sort of quality control metrics. The normalization had huge effects on all sorts of the CPG sites. And we noticed that for the first generation and second generation clocks, the normalization schemes across those CPG sites were pretty consistent. They kind of went along sort of a diagonal of both were making the same prediction. For the causal clocks, it seems like they kind of just picked CPG sites. I don't know for what reason, but the ones that they picked happened to be very noisy. They tended to have lots of failures in HRS. They tended to also be extremely affected by normalization, which means they're probably ones that tend to be towards 1 and 0, because those are the ones that really get hit hard by normalization. So I think what's happening is that, especially, so Cathal, when you did time 1 and time 2, they were on the same plate?
No, they were
Okay.
they were done at different times, because the initial batch had been done in Italy.
Okay.
Yeah, but I adjusted for chip type
Yeah,
in the analysis here, in addition to, we use select probes, common epic one.
Yeah.
We also adjusted for the chip type in that analysis.
Mhm.
Yeah.
because I'm wondering, so we're often interested in epigenetics because we think it changes and it's interesting when it changes. But there's a whole group of people who are interested in the metastable epialleles that do not change across the life course.
I listen. My departing gambit is I think we've dispatched with the first generation clocks.
Sure,
So,
yeah.
Yeah.
I think the first generation clocks, age is still the best aggregator of damage.
age.
Yeah, yeah.
So and we kind of dispatched with those clocks.
But I think there's a lot of work that we need to do just throwing out CPG sites that
I
are.
wasn't sure how we
just
did
noise.
time. Okay, there's more questions. Sorry, I didn't mean to run away. I'm very happy to answer more questions. I was just conscious of the time.
Data clocks are not trained with lasso, which will get rid of all this.
They were then in the second step, right?
I think so, but they had
The first step was the just
MR.
feature selection,
Well,
right?
yeah,
The
but it was
MR.
based on
Mhm.
Mendelian randomization. So
Mhm.
the primary underlying
Yeah.
training is not to make the most predictive model.
Right.
It's meant to be the most. So I think it's a very good idea, but I think how you implement it.
Mhm.
Technical thing.
Matters. How you
Yeah,
implement it
The
matters.
MR might have even picked less reliable probes.
Yeah.
Because they had more variance,
Yeah.
because they're bad probes.
Yeah.
And so they chose a procedure that generated a clock that has a lot of noise, essentially.
Interesting.
I was running away because this is the realm of the molecular biologist rather than testing.
Thank you, we're gonna continue.
So I thought about presenting all of the work that our group's done on neighborhood and epigenetics, but then I decided that it would be nice to take advantage of this particular crowd to instead talk to you about a project we've been working on for years and see if we can crowdsource some thinking on what we're doing here. So I work on this project with Eileen as well as Eric and EunYoung. So I do all my work on neighborhood environment exposures and health and aging across the life course, trained as a sociologist, demographer. I think a lot about upstream factors. My factors of choice are all of these different aspects of the physical, social, built, chemical environment, managed environment, natural environment, and also their interactions. And then these are, the outcomes that have been studied in this literature for decades: disease endpoints, disability and frailty, mortality. But between like an environmental exposure and a health outpoint, there are a lot of biological steps that occur in the interim. And so trying to understand some of that biology and identify some of those underlying biological mechanisms would be really helpful for the literature to understand this question that I learned about when I was in school, that, again, Teresa was one of these people, but like, how do our environments get under the skin, and became like such a catchy phrase. It was sort of cringy, and I've circled all the way back and I love it now. But I think like if you're thinking more of like basic science, perspective, it is really, it is even hard for me to understand, like, how do we leap from this thing in the environment all the way to a major disease outcome, for instance. And so this is where we're trying to do the work. And this also happens to be the entirety of the current focus on the exposome, which is trying to understand how your lifetime of all of your exposures and experiences interact with your biology to produce differential aging outcomes. So there's quite a bit of research that's been done on a lot of different air pollutants. I focus mostly on PM 2.5. This is one of the nation's 5 criteria air pollutants. It means that the EPA monitors for this pollutant, it regulates this pollutant, and it's because of its known ubiquity in the air we breathe, and also its many adverse health impacts for populations. So many studies in the United States and other countries around the world have shown impacts on multiple organ systems. So there's cardiovascular impacts, respiratory impacts, gastrointestinal, a lot of impacts on the brain, particularly the developing brain and the aging brain. This figure on the right side is from a review that was recently published, where they basically trying to make the argument or have come to the conclusion that epigenomic damage is the most plausible biological mechanism of air pollution derived carcinogenesis. So they're really pushing this idea, like, but you know, they have a lot, this is very convincing. So on the right side here, they summarize kind of all of the biology we understand at this point from human studies, animal studies, and in vitro studies. And it's compelling. There's a lot of really solid research showing that there's oxidative stress, vascular alterations, inflammation, impacting mitochondrial functioning. And so, you know, we think that maybe what pollution is doing is accelerating aging. Again, I'm gonna bracket your point earlier about what we actually think aging is, but if we're in this realm of what, you know, the way NIH and Geroscience have been talking about this. There is some kind of promising signaling as well. So this is a study from a few years ago where they were trying to identify CPG sites associated with prenatal exposure to fine particulate matter air pollution. You can see that they found 14 CPG sites that were associated with this prenatal exposure. And then this is a similar study that was done, but in that MESA cohort that Lauren did her validation work in. They found 5 CPG sites. None of these CPG sites overlap across the 2 studies. But so there's, again, kind of sort of making sense. And then we go into the actual studies, and we've done a review. We've done about a half a dozen studies that have tried to establish whether there's an association. And the main takeaway message in this last column, I've tried to highlight in red what the associations are, and the non-significant associations are in black. And you can see that it's, I mean, I think mixed evidence is maybe a too strong of a word, it's maybe no evidence, but the problem is we don't have that many cohorts where we have these pollution exposures and we have the DNA methylation. And so you can see that the cohorts here are really the normative aging study, which is a really good study for chemical exposures and aging. But these are older men associated with the VA system in Boston, and they're mostly white. There's also the CORA study, which is a study of older adults in Augsburg, Germany. I had to look up where Augsburg was on a map because I'd never heard of it before, which is no knock on the study. And then we also had a sister study, and this is a very kind of strange study if you're thinking about a population like a probability-based study, but these are sisters of women who have breast cancer. And so like, you know, these studies are not necessarily ideal, but there was a study published just 2 years ago that came out of the health retirement study. Okay, so the normative aging study, they found an association with the Horvath clock, but not the Hannum clock. This next study, this other study in 2016, compared these 2 cohorts, and they did actually find associations with the Hannum clock, but only with intrinsic epigenetic aging. When they looked at the extrinsic aging, there was no association. I did put here that they found something with black carbon that'll be relevant for later. The sister study found no associations with Hannum, Horvath, or Pheno. The normative aging study folks then later went in because they had gotten the pheno age clock. They didn't find an association with pheno and particulate matter, although they did with lead. And then the sister study came back with their next set of clocks. They don't find anything with DNAm, with germ age, with pheno age. And then this health and retirement study paper only found an association with the POEM clock, which was all they had available at the time, but they also only found it for the non-Hispanic Black participants. And so this seems like a bit of a hodgepodge and not very strong evidence. But nevertheless, we thought, let's just check it out, HRS with all the different clocks and see what we can find. So we used the DNA methylation sample, it's basically the same sample Lauren was just talking to you about, and then we have the tracked area level concentrations of fine particulate matter air pollution where these people live. This is data that we get, that we kind of massage a little bit that comes from the EPA. So this is, I always say, the most null findings results slide I've ever put together in my career. But this is all 5 of these clocks. The dots represent the point estimates, and then they just have little confidence intervals. And you can see that none of these associations are statistically significant. They're not even appreciably really different from 0. If you look at like the germ age clock, they're all essentially 0. We found the same null findings for black carbon and for lead, even though I showed you in the prior slide other studies had found associations. These models are really well controlled for all of these characteristics that we normally control for in the models that are listed at the bottom of the slide. We also tried to understand if there was stratification by age, by sex, by race ethnicity, by region, and then you can also see the different markers on the vertical axis of these graphs are different exposure windows. So we've even done a much more extensive analysis of exposure time than any of the other studies had done, ranging from the 7 days prior to the blood draw, all the way up to 10 years prior to blood draw. And there's no association across all of this. So we think probably, that was a strong statement or conclusion coming out of this Frontiers paper that was published last year. However, because we're working in this group in the biology, as someone who's been doing most of my work over my career on the environment, I have been thinking a lot about how can we leverage our biological measures to actually get better measures of our environment, which is basically what Lauren was talking about earlier. And so I think a lot about this, particularly with air pollution. So this graphic is kind of adapted from an EPA graphic, but if you think about our exposure here as particulate matter air pollution, we get that into the body primarily through inhalation, so it's coming in through your nose or your mouth. There's the exposure, and that's what I just showed you in the slides. That's what we're monitoring in the outside air we're breathing. But what that translates into is something called a potential dose. So that's what we say that's what people are exposed to, but the reality is it's what you could be exposed to, because I don't know how much time you spend outdoors. I don't know how solidly sealed the windows are in your home. I don't know if when you drive on the freeway, you let air circulate from outside because you're worried about carbon CO2 buildup, or if like me, you always hit the button so that you don't get any air in, particularly off the 5 freeway, which has been shown to have very high levels of air pollution. Okay, so you have your potential dose, which is whatever we're measuring outside, and that whatever we're measuring outside is primarily what we tend to have, like that is how we're measuring these things. There's also something called the applied dose, which is like you breathed in particulate matter, but we would really ideally want to know like how much of that actually made it to your lungs and wasn't caught in your, the lining of your nose and all of the other different systems we have in our bodies to actually make sure that the external world doesn't get inside. And so we would call that the internal dose. But even after something gets into your body and theoretically, let's say we could measure your internal dose, there is also something called the biologically effective dose. So this is basically, what is the dose that your body was not able to clear out, that we wouldn't see measured in urine or some other tissue, but like what is the dose that got into your circulating blood, that passed the blood-brain barrier, that embedded itself into an organ in your body? And that is probably ideally what we would like to measure, and it would be much less noisy than just getting a monitor-based measure. So we were looking through, some of the work and thinking about how we could use something like a polyepigenetic score, for instance, to try to approximate the impact of this PM2.5 exposure on the epigenome. So for people who don't know, and I think it's hilarious that I'm gonna explain it to you because Eric was the one who had to explain it to me, but these are derived from these EWAS findings. Basically, those two plots I showed you before, those Manhattan plots, we would just go to a study, find the CPG sites where they said that they had, highlighted DNA methylation that was associated with whatever the exposure was. You saw Lauren just did it for a very different kind of exposure, a social exposure, but you could do this for any exposure. And so we're thinking, the theory, and this might be a strong assumption, is that this could summarize like your cumulative environmental impact on methylation. And that if the concept of a polyepigenetic score is unfamiliar to you, it's very much like a polygenic score. So it has the same kind of thinking. And so it just aggregates this genetic risk and it's just trying to quantify that contribution. So when I say we, I'm always referring to Eric Klopak, started this process where, he went to the EWAS Atlas. And he went and grabbed this study. So this was the normative aging study. It's the one that I showed you before in the table. You might also notice this is the MESA study I showed you the Manhattan plot from. We actually decided not to use that study because of exactly the things that Lauren just told you about, and I'll talk to you about that as well. But we were trying to get studies that could align as well as possible. So this was the process that we used, which was, so Eric just went in and I helped him try to, understand this stuff too, because this part was new to him, but we're really trying to understand the methodology and the sample characteristics to make sure we have good enough alignment to actually do this exercise. So we were looking at the exposure. In this case, it's an interquartile increase in PM2.5. That's fine. They have very specific exposure window, but okay. Population characteristics, like I said, it's men. It's not great. They're all like 70 and older, also not ideal, and they're all in Boston, but close enough, I guess. This was the reason we didn't go with the MESA study, because they were in monocytes, and we were like worried that if we went outside of the tissue, that would give us a problem. And we also were worried about this, which was, as Lauren points out, because they used a different chip. So Eric went ahead and he did this process where he grabs the information from this EWAS Atlas. He gets all of their CPG sites that they found. He then grabs the HRS probes for each respondent. They're indexed in this equation then that he uses to calculate a score for each of them, which is just weighting all of their CPG sites by the weight that we got from the normative aging study. And so from that, we end up with a score that we can then plug back into our models, in the hopes that this might be approximating more of the kind of biological uptake or dose. This is when we first start to notice that things have gone awry. As we would expect, there was a high correlation among the pegs for PM2.5, black carbon, and lead. These correlations among the pollutants for me made sense. The ones that were stronger and less strong, everything made sense. Here's where we got a little confused because the polyepigenetic scores for these pollutants had no correlation with these like outdoor monitored model-based measures of concentrations where people live. So that was our first sign that maybe this wasn't going to work out. Then we went back into the models. We added measured levels of PM2.5, lead, black carbon. We also added this PEG score, similar to how Lauren did it, put it all in the model. But this was interesting because previously, when we were looking, in this case, I'll start with the pace clock, but you can see the measured for PM2.5, black carbon, and lead all say that they're not significant, but we did find a hypothesized expected positive association with the PEG score, and they were statistically significant. So then we move on to a couple other clocks looking at PM2.5. Yeah, looks like it should for grim age, looks like it should for pheno age. Then this happened, and now these are negative associations, suggesting that more methylation from these pollutants is associated with decelerated aging, which is obviously totally counterintuitive and contradictory to all of the biological findings. So we're not sure like exactly what's happening here. And then we sat down to really think about what we had done. And we realized that maybe it's problematic to use a PEG and then use it to predict a clock. So when we make a PEG based on these CPG sites, it's possible that there's going to be some overlap between that CPG site that we made for the exposure and whatever's being fed into the modeling of the epigenetic clock as well. And so we're worried that we ended up in a little bit of a circular reasoning here, and there was probably a lot of high collinearity. That could explain the weird negative associations potentially. But the other thing that I didn't like in particularly, and maybe just being a demographer, was like, you know, the pollution environment in Boston is very specific. We took it from a very highly specific geographic area. We're trying to extrapolate across the country, and the sample characteristics are super specific too. And I do think when we make some of these biological measures, we always have to be worried that if we're making them in like a very like European-derived population, for instance, and then taking it into a very race ethnically or socioeconomically diverse sample, that they're not always going to function very well. So our big takeaway is that we're worried that this is a co-associated process and that it's probably not the direction that we want to go in. And as Eric pointed out, we just stole this idea from genetics and assumed it would work, but maybe it's not really what we want to do. So there's lots of problems, but we like Eric because he always comes up with new solutions. So he says, why don't we just develop a surrogate? So instead of selecting individual CPG sites that came from some other study, that we can actually use a machine learning approach, which would be more flexible, and we'll derive this composite DNAm measure using thousands of CPG sites instead of like the 5 or something that came from the normative aging study. And so there are a lot of advantages to using the surrogate approach instead of a PEGS approach, particularly that it will optimize all of the CPG sites data instead of being limited to whatever another study found. I do think being able to capture nonlinear effects is important. We have people in our groups that are doing some machine learning models on nonlinear effects, and it seems to be working out well. And then also, we can adapt these to different populations, so we don't have to worry about this concern about not being able to generalize across cohorts. The only downside I can see is that the surrogate approach is we're going to lose that ability to get that specificity in the CPG sites. Although I will say, all those CPG sites I showed you before, some of them are related to things like asthma, respiratory problems, cancer pathways. There's like protein signaling that maybe makes sense. But like half of the CPG sites that have been identified in these studies, their biological importance is totally unknown. So I don't know how much of a negative this is. Okay, so having said all that, we think this is our next step. However, when we were kind of thinking about what is it that we need to make a good surrogate, we realized, as Eileen said, first we need to find a good and reliable friend. You see what's happening? Good and reliable, some criteria here. Or because I just saw that Lauren's validation in Mesa didn't go too badly, maybe that's another good, reliable friend. Since it didn't seem like it really was too big of an issue that it was a different tissue and a different array. But yeah, we need, comparable data. We're saying same tissue ideal, but not necessary. Eileen's been really pushing through the center, her work in the center on aligning pipelines, because as we're hearing, everyone's doing everything very differently, and then we have to be able to coordinate across study teams. So this is a direction we'd like to go in. I have no slide after this because there's no point in us doing this if we don't have someone that we can work with to validate this in another sample. So this is the concluding slide with like a please help me face.
You had him at good and reliable.
I said good and reliable, and he started looking behind him.
I thought it'd be facetious. Yeah, we'd be happy to help out with that.
We would as well. My student is doing this in Airway, which is the cohort I showed you earlier.
Great.
I would just say two things. PM2.5 is just the measure of size. It's probably very different in America and Ireland.
On different places in America.
Yeah. So I don't know if you really expected to elevate. And also, what was your resolution? Because I don't know what a tract is.
Oh, so we use a census tract. It's a resolution of people, not size, but it
Yeah.
ends up being the equivalent of maybe 4 to 6 city blocks, or if you're in a lower density population area, it can be a broader geographic area. But we can actually get all the way down to a much smaller resolution, more of like a block.
Yeah,
So,
I think you probably should
10 by
try
10
and do that
kilometers
if
or
you're gonna
something.
try and do an individual level.
And we can also do source fraction on PM 2.5.
We might actually be a better cohort than you think for that, because like in Ireland, particularly in rural areas, a lot of people still burn peat. And it's really toxic, right? So there may be quite a lot of exposure actually to that. The government have actually recently legislated around you're not allowed to dig turf anymore, and you're not allowed to sell it.
Really?
Yeah, in Ireland, yeah, and it's been really controversial. Yeah, but we've wondered about this before, because about how to measure. We talked about remote sensors or the
Yeah.
silicon bracelets. You know, you can pick up exposure for people, because you know what the area level exposure is, but you don't know what the person's being exposed to in their own home. Open fires, but in rural areas, it's still really prevalent. Open fires, peat burning, and so on. So there may be a lot of variation across our cohort and exposure.
Yeah, I mean, the pollutant variation bothers me a little bit too, but I can't be too bothered by it because we have a lot of variability within the United States too. So like the things that are in PM 2.5 in Los Angeles are probably different than in Boston and different than in New York City and Spokane, Washington, and the Rust Belt in the southern United States. So, I mean, I think it's at least worth it. It's not any different than doing like SES though, like social environment
But
is
I would
also
have
not
said,
that
I would have said
comparable.
the same about SES. I mean, it means something different everywhere.
Yeah.
doesn't it?
That's interesting.
It just doesn't replicate air pollution on methylation. I mean, there's been a lot of studies and I haven't seen any replication at all at the CPG site. That's not to say this isn't going to work, because my student's doing this and he's going to be very disappointed when he sees the results. So
It's my mission to let people know not to let their students waste time on this because we've wasted a lot of time. The other thing I did want to mention, I think I forgot to say, is the pegs that we made might be fine if we're predicting a different outcome. Like this was very specific about going for clogs. But I think what I would still like to explore the idea though of getting better measures of environmental exposure, in particular these kinds of things where the best we can do, I mean, we spend so much money and time and effort in many countries trying to get very precise estimates like at your doorstep, or maybe putting like personal air pollution monitors, like little cat collars essentially. And that still is not really telling us, each one of us is gonna have a different ability to deal with an environmental exposure. And so it's really just trying to rethink maybe the way we're doing exposure science and see if we can leverage the biology at all. Maybe it turns out we can't, but I think it would be, if we could, it would be worth trying to see.
no, I'm a complete believer in this, the exposome approach, but I'd say it's going to be challenging.
So, I mean, I actually think it's really good to try to dig into this a lot more in part. So that neonatal study that you mentioned at first, so we took those and created those pegs in a few years ago in Future of Family. So that's the child cohort. And we couldn't find it for 2.5, but we found it for 10. And we saw that signal 9 and 15 years later, even adjusting for current pollution exposures, which also makes me think if there's, I don't know how long that signal remains, but like it kind of makes sense that there might be a stronger signal of, say, in utero exposures. And I don't know if that, I mean, that's something you could calculate pretty easily in HRS, but I think ultimately Eric's right that like trying to train this at EWASs are not really designed for creating predictive algorithms. They can be used that way, as you're saying, but like they're not going to be quite as good. So I do think it's a really good idea to try to dig in and maybe find a really well characterized sample and then try to replicate it into HRS actually. But like, I do wonder how much of this is, again, going back to what we were talking about with Lauren, is the timing of these things and how much of this changes over time. And is this like a chronic exposure? This might be one of those things where you know, a life course model of exposure rather than like a time point might be a more appropriate thing, and taking like more extreme exposures
Mhm.
to help you really tease out, because I don't know that it's gonna be able to be really fine grained.
Yeah. No, that's a good idea.
Yeah, I just wanted to follow up, and we haven't been doing too much work on air pollution, but we've been looking at lead and these epigenetic surrogate markers for lead. Um, But we've actually measured in the high school and beyond study, we've measured 9 metals and we have DNA methylation on everybody. So our plan is to try and create these DNA methylation surrogates in high school and beyond, and then try and apply it to HRS to see if that can be done. I think that would be a nice way to replicate some of those things.
Yeah,
Right.
we, cool stuff. The Excel sheet you showed, I think that Eric used to create, you know, which markers to include. One general point is the clocks are really great, but looking at the individual genes, you can still learn a lot. Like if you
Yeah.
look at C2ORF
Mhm.
78, you look at C18ORF 20, they're not named. So the likelihood is they're not characterized. So even though you can't maybe validate these clocks or these algorithms for noisy reasons, those loci are being selected for some type of pattern, even within the noise. So this one general observation I would say is to not ignore those and like make note of it and look at the history of those genes, because they may have important biology, even though you can't actually, you know, replicate this in a different cohort. It doesn't mean you should never even study this, because replicating is going to be challenging no matter what you do because of the noisiness of human population data. So if we ever see like genes like this, I think we should actually look into why it hasn't been characterized and make note that maybe it is involved in whatever like biology that you're seeing, whether it be air pollution or some type of epigenetic regulation. Just like a general comment, if you ever see stuff like this, just make a note of it and look at the history of the gene, because it might be worth characterizing and figuring out what the protein or peptide is doing. Like the C10R78 is a very small peptide and there's no studies on it, probably because it's very difficult to make.
Yeah, so like when I looked these up, I was like, okay, this one's like a respiratory one, this is a cancer one. There was one on like, it was developmental mental disorder, I didn't understand that, but maybe it had something to do with the brain. And then there were some that were like, this is a peptide.
Yeah, I mean,
That's
like,
all I could figure out about
like at
it.
least for me, the assumption was, oh, all these genes are characterized. We all know what they're doing. And the reality is for a lot of these small ones, you just don't know.
Yeah, but I think
you
under.
guys like the known unknowns.
Well, I-
But there's-
We focus on
there's-
no.
These,
but they're
this
just never
atlas,
characterized.
this atlas, like this is all coming from population studies work.
Yeah.
But I did wonder when I looked at these, like if we just circulated these to our biology friends, would they like you be able to say like, yep, we know what that one is? It's just that not everything you guys are doing in the biosphere has, you know, made it into the population.
Yeah, it's like you're seeing really cool patterns and signals, right? But because they're not known, naturally, like trying to figure out what they're doing, it's not an assumption, but maybe it's just overlooked.
Mhm.
There's no function to it, but that's still important as well. Whereas for like biologists, we look at the ones there isn't function, but we don't do stuff like this.
Yeah, but I
right?
like Lauren's idea of maybe trying to, like, this is a very a priori kind of approach, and then the machine learning is this like total blind box algorithm. It would be nice if we could have like a, I don't know, isn't it like Tesla drivers, like it drives your car, but you have to like keep a thumb on it or something, something like that. And then to Colter's point, I do think one of the problems with these studies where they've tried to identify the CPG sites is they don't necessarily do it for the extremes of exposure. And so that might be noisy just because of the way that they're doing this work.
Just as a quick, I know we're almost done. It's just, keep in mind that these chips are not representative of the entire methylome.
Right.
They are actually mostly selected for cancer research. So it's not surprising when you look things up, you're like, oh, this CPG site is related to cancer. That's really interesting. Well, yeah, that's because of 60% of the probes are intended for cancer research.
Okay. Thank you.
Our next speaker is Colter Mitchell.
Oh, come on.
No,
Higher skip to the gab is keeping us,
Oh.
there we go.
Everybody's anxious
Yes.
to hear this.
All right. Hi, everyone. Colter Mitchell. It's nice to be here. Thanks, everyone for putting this together. Let me talk a little bit about DNA methylation and harmonization. So as you kind of already
You're
heard,
gonna be a biologist.
DNA methylation is used in a wide variety of applications. It's kind of like why we love it, right? It can be used for creating new stores, pegs, as you heard earlier, for doing site-specific DNA methylation analyses. And we use it in all these different ways. And one of the nice things about what we do is most of the population studies use Illumina arrays. The 450K, the EPIC 1 or version 2. And as you've already been hearing, we all try to use similar QC pipelines. We try to use the same algorithms to create the exact same clock so we're all on the exact same page. But does this mean that we are actually harmonized? It's a great question. I'm going to just put this up very briefly. I'm going to say a lot of different words. When I say sample, I mean like, you know, it's a blood sample or it's a saliva sample. Plates, you'll hear that. So that's what a plate looks like. You have a well in a position or on a plate. You have arrays. You have chips and slides. So the big things are a slide or a chip. You have an array, which is an individual. On the 450K, you have columns and rows. We won't talk about that anymore. So each array has hundreds of thousands, almost a million in version 2 of probes, CPG sites. Each probe has many beads. I won't get into what that is, but it's a QC thing that you'll hear me say briefly. So like 20 to 40 beads for every probe that is measuring methylation. A big thing to keep in mind is when I say batch, what I mean is that's either a set of slides or plates run at the same time, usually on a given day. So we'll call that a batch, right? So you've heard people say batch. That's usually what we're talking about. It's usually a set of plates. And the other thing you'll hear me say is technical replicate. What I mean by that is it's the same sample in the same batch, at least, even more often on the same plate. So that's a technical replicate. And then you hear me say duplicate, which I won't, but I often use it in other things. It's the same sample run on a different batch. Okay, so after saying all that, why have you been hearing the word batch effects? There's actually a lot of work in methylation on how to adjust for batch, how do we account for batch effects, what are batch effects. So there's been a lot of attention on this. Even in very well done studies, it's not uncommon for it to explain 10 to 15% of the variance in methylation data. And I've seen it explain up to 30 to 40% of the variation. Sometimes people will try to do things like randomize. It can be really problematic if you run a batch of cases and a different batch of controls, because then you are perfectly confounding those things. So people have to be really careful about randomization. HRS is a great example of this. I remember when Jessica was laying these out and she randomized everything and then checked on several covariates to see if it still looked okay. There is still a problem is if you have a study with tens of thousands of variables, by chance, one of them is going to be related to batch. And you know if it's a effect of that question or batch. So that's often a thing that we have to just kind of accept. The other thing to know is that most longitudinal studies are on different batches. We're not going to talk about that today, but that's a different thing as Cathal was describing. And you might realize that if you run something in a different lab, kind of by definition, that's at least a different batch. Okay, so what we're going to talk about today is an experiment that we have the data for. 5 sets of 48 samples were sent to 4 different labs. These are the labs that ran the methylation data for LOSI, for HRS, for Nicola, and for Tilda. So this is, even though I'm the one presenting, there's a lot of people in this room who have been involved in this. I said 5 sets, but I only gave you 4 labs. That's because one of the labs, lab 1, had 2 sets. So one they kept in-house and then just ran it. Another one they shipped out and shipped back. So it would be like they were shipping it to themselves. So that's kind of our batch effect. And we have a lab effect. The one thing that's a little bit strange is lab 2 and lab 4 have Epic version 2, and the other 5 sets were Epic 1. So I'm just going to talk briefly. I'm actually not going to go into the QC protocol, but the first thing that we did for this was just run it using the HRS quality control pipeline. So I'm not going to go into all the steps of that because that's actually something we hope to do later on, is testing different versions. But that's the first start. So if you want to go and read HRS, you can see exactly what we did. We followed it as closely as we possibly could. So you heard before, new normalization, quantile normalization was not used. We also had technical replicates on all of these, on most of the samples. And this is thanks to Eileen and Jessica, who have this RO1 trying to examine, and Cathal. So NIH is through Eileen and Jessica, and then we have Tilda and Nicola, so Cathal is a big part of this too. So the 3 countries are US,
And Lassie.
Ireland, and then yes, Lassie from these other sets. Okay, so I'm gonna go through these very quickly, just so you have a sense of this. But now you know about the experiment. There's no substantive interest here; it's purely methodological. But we'll look at some QC metric comparisons. We'll look at cell distribution estimates. We'll look at smoking estimates, blocks, and then we'll actually get to a EWAS. Okay. So I'm going to show, the first thing you should know is all 240 samples that should based past QC. So they all would have been incredibly high quality samples. They look really, really good. But you can see each of these colors are a different lab. Remember, these are the exact same people. They do not overlap perfectly, right? So you can see, like, here's one lab. This is one of our Epic 2, I think this is lab 2, stood out, had a lot higher intensity.
You're masking the labs.
Yes, I'm masking the labs. If people want to know, I can tell them later on. I was informed by your co-PI that I needed to mask all the labs. So there is one lab, you might say, oh, it stands out. It's unique. So that we'll come back to that in a little bit. In fact, we did this on 17 different Illumina control metrics. Again, all of them passed just fine. We wouldn't consider them problematic. It was not uncommon to see one lab stand out on one of these metrics, to cluster differently from everyone else.
Was it always a different lab or was it always the same?
It's not the same lab. It was often different. So this is a different one.
It was better when they had their names on them.
Well, yes, okay. If you really want me to, I can reveal who that was and who this is.
No, you don't need to.
But they all pass, and that's one of the things that's interesting. One thing that's particularly interesting is after we did all those, we also checked for sex, for sex. And here we had, you'll start to notice a Y chromosome difference. So, and that's actually an Epic 1 versus Epic 2. So Epic 2 just has lower Y chromosome intensities. And I haven't seen other papers report, but I've talked to other labs that have run it and they see the same sort of thing. There's just a lower intensity Y, but again, still very clear. I don't think there are fewer probes, but it's definitely a lower intensity. And slightly different for X, but it's not that big of a deal. So if you just start looking at the comparison of the cut probes, if we were to go through our regular QC process, you start to see differences. You have the lab one was shipped and one was not, so that's our batch kind of difference, and that's like pretty much perfect, same number at least. And then you have lab 2 and lab 4, and those are our Epic 2, so they different numbers, but similar percentages. And one of the things that we saw very quickly is that the Epic 1, around almost 6,000 of those cut probes were the exact same across all 3 plates, and around 17,000, so almost all of like lab 4's version 2 were also in lab 2. That's the version 2. So, what do we make of this? Part of it is, I think, to keep in mind that which probes get cut is a little random by group. It's the exact same samples. Keep that in mind. And this is a distribution of all the different studies. And without normalization, you'll see that there's one that kind of is slightly skewed left. This is one of the labs. This is an Epic 1, and the rest kind of line up okay. Okay, so let me give you some specific examples. So estimated cell distribution. This is one of the primary functions of DNA methylation is cell differentiation. And often when you run it, if there's no experimental problems, the first PCs of methylation data are related to cell distribution. So the other thing is a cell, and you saw this on many of the papers, cell type estimates are regularly used as confounders, right? We're worried about that. That's really what's driving this, or potentially as a mechanism. So this is a really important thing to get right. And so we did this with a couple different versions. I'll show you one. So we did EpiDish to estimate cell distribution. So it has a wide, relatively large number of different cell distributions it estimates. So we heard a lot about T cells and B cells yesterday. Yeah, we had that, we estimate those. Here's the thing, is actually it was incredibly precise for these, comparing across all of these. So that cell type estimates were within 10 to 15%. Remember, most of these cell type estimates are actually like 3%, 4%. So it's actually very, very close and highly correlated. The one exception is monocytes. For whatever reason, it was much harder to estimate those. And in terms of correlations, on average, the cell distributions correlate about 0.92 to 0.99, so very highly correlated, except for monocytes, which were 0.78. I think we've talked about this. Monocytes are often a little problematic to measure, but
Did you use the monocytes from the complete blood count?
yeah, we've compared it to that too, and they compare pretty well. They're far more correlated with each other than they are with the actual measures, but they're still highly related. So overall, pretty strong concordance. Let's talk about smoking. And I'm running through these because this is not the exciting stuff. The next largest association is often DNA for DNA methylation after cell distribution and sex is often smoking. Smoking, including maternal smoking during pregnancy, leaves a pretty strong signature on the methylome. So within our sample, we had 17 current smokers, 14 former smokers, and 17 never smokers. So keep that in mind. And we used EpiSmoker, which is a robust classifier to determine smoking status using DNA methylation. We have other ones too, and the similar results. So here's the short version. Of the 17 current smokers, all labs, batches, and technical replicates found 14 of the 17, and most of them found 16. So there's a handful, a couple that they all struggle to find, but in general, they do a pretty good job finding people who are currently smoking. Similarly, never smokers, all the labs, batches, technical replicates, all said 15 of them, and most times 17 of them were never smokers. So it did a pretty good classifying them. The 14 former smokers, however, it was really a coin flip on whether or not they were able to determine if they were a former smoker, so they would misclassify that group pretty regularly. The technical replicates do a pretty good job matching each other. So that's the ones on the same plate, but like once you go to a different batch or it struggled with former smokers. I was not expecting this. This was a surprise to me that it was so poor at finding former smokers. Also, people could be lying. Right? So there's always that part too.
But then they would all be
They'd be all
missing the
flying
same
everywhere.
ones,
Yeah,
right?
I mean, but that's the current and the never smokers is for sure. I don't know, some of those might be that way. Yeah, so the technical replicates matching the former smokers 87%. But if you look at like general smoking probabilities, those correlate from about 0.88 to 0.94, but I think that's mostly driven by the current smokers and never smokers. Okay, so this is what we were running through to get to kind of more interesting things like clocks. We're just going to look at 3 clocks in this case, kind of a pace, you need pace, pheno, age, and Horvath. So kind of a range of different clocks. This is, remember, exact same people. We should see the exact same results across all of these. Should be the same age. And what you see is that they're not, right? Pretty different. We're going to show some others. We'll come back to this. And you see that the Epic 2 are slightly different than the others. You can see that the shipped one, so lab one, stayed pretty close to each other. And so that's kind of that batch effect. So you can start to see some of these differences. But I could spend a lot of time going over this, but we're going to show more versions of this. But these data that make me over and over again say, this is not ready for prime time for taking it to someone and saying, this is how old you actually are.
Yeah.
So just as a sense though, like if you take pace, you look at pace, this is just a correlation, and they're pretty highly correlated, right? 0.96, for example, for a lot of these, for like the Epic ones are pretty highly correlated. Epic 2 is not as highly correlated as Epic 1. But they're not bad. They're not bad in terms of relative. We have, and I did this last night because I realized the ones that we had that were more similar actually just still had age in it, their estimated age, and they look much better when you have age in there because they all share that. So if you take age acceleration, they're still not bad. So that kind of upper cluster right here is Horvath, age acceleration among the Epic 1 group, and that's not horrible. So that's already talking about the residual. It's not quite as good when you look at like the Epic 2, but it's not horrible. Pheno age is much worse, especially for the Epic 2. But I think we've kind of seen that more and more. So let me just give you a couple examples of what this looks like. I like doing this. Other people hate this plot, so you can tell me what you think of it later on. This is their actual age. This is not age acceleration. I'll show that one in a second. The black dot here is their actual measured age, and this is their range of their different estimates for their Horvath age. And you can see some people are higher or lower, and you can see like that would be kind of the age acceleration, which I'll show right here. So you can see people are potentially picking up on people are slightly higher, people are slightly lower, but look at that range. Right? And that's a key thing. You can see multiple color, the same color dot. That's a technical replicate always. And you can see some of those technical replicates are very close, others are not. But this tells me again, like it's getting general ideas. It's still not precise enough. So this is pheno age. And yeah, this is like we get strange ones. And this is what this looks like. And yeah, you try to, if you stare at this long enough, you think you can see different things, like there's two greens, but that doesn't really tell you anything. Yeah, so we have that. This is pace. Again, what pace does that's a little tricky is that because it's such a narrow range to begin with, like for this one, if you take this one out, we're talking about from 0.7 to 1.4. And then when you start to realize like how wide this is like a third of the distribution, which is not nothing. So just as a general sense, I put this at, this is the pace, this is like the rate of aging from 0.6 to 1.4. Horvath age acceleration from -11 to 18 years. Pheno age is -25 to 38 years. So what would this look like? Within the replicates, this is like it's pretty good. The replicates suggest, and by the way, this is the thing that I often see when people report this, is they'll say, yeah, our age range is like 3.2 years. Like that's your technical replicate one, which is actually it's not bad, but that's because you're running it on the same plate. Once you send it off to a different batch, for example, for the Horvath age acceleration, it's more like 5 years for the range. And for a lab, it's like 6 years. For pheno, it's larger. So, you can get within 6 years if you send it to a different batch or a different lab. I don't know. That doesn't feel precise enough to me to then determine like what your health action should be. Okay, I've kind of blown through all of that. Just wanted to quickly end with a fun thing, which I'm still trying to think through, which is we actually did we calculated coefficients of variation. So this is the standard deviation divided by the mean for each individual at each CPG site, right? So they have these measures, and I'm saying, let's just pretend that that CPG site is a real thing and we have variation for an individual. So this is a within person coefficient of variation, which is tricky. I'll mention the other types, but this is a within person variation. And just so you know, methylation as measured kind of at a population level is mostly invariant anyway. Remember I mentioned that this is CPG sites mostly focused on cancer. You can already see that just if you look at it from a population perspective, coefficient of variation is quite low for most methylation data. So there's just not a lot of variance. There's a handful of CPG sites really that have a lot of variation. But within person is even less, and that's actually a good thing. So low coefficient of variation, within person coefficient of variation means that you get roughly the same measure from each time. And so this is good. It's good that it's skewed far left, closer to zero. But you can start to see that there are some CPG sites out here that have more variation. And really, in order to even calculate a reliability, to have enough variance for reliability, you need a coefficient of variation of around 0.25 or 0.3. So anything less than that, there's so little variation, you can't even calculate a reliability. So we did an EWAS of this within person variance. I don't know that anyone's ever done this before. I've done it with other similar sorts of things, but this is kind of strange. So this is an EWAS of that. And you can see, even though there's not a lot of variance, we still get some significant variance. So around 24,000 probes were significant at kind of that standard cutoff. And I don't necessarily believe that that's really important until you start getting at coefficients of variation greater than around 0.25. So that's an arbitrary cutoff, but I did that. We have around 8,937 probes with that. So these might be ones that we start to explore, seeing whether or not these are batch influenced or lab influenced CPG sites. There are 30 probes that have a coefficient of variation over 1, which is a significant amount of variation for methylation data. Okay, so kind of gone through a lot of things very quickly, hoping that we can have good discussion. So lab effects are slightly larger than batch effects, but in some ways they're similar. You can think about it that way. And those are larger than we can see with chip effects, with a few exceptions like the Y chromosome. QC differences may seem large. I didn't go into this into a lot of detail, but we actually tried to take all those QC metrics and predict like differences by clocks, differences and other things. They may seem different when you're looking at, like, there's that purple lab over here, there's a higher intensity. That doesn't seem to translate into major differences. The cell distribution estimates are very consistent, both in absolute and relative terms. Smoking seems to do pretty well with current and never. Former smoking, I think, is a mess. I think it's just tough. And I think that's partly because you don't know whether they're a former smoker 30 years ago or a former smoker last year. I think that's where it gets challenging, and we didn't get into that detail. The clocks, I would say, have high relative concordance. They're correlated fairly well. Again, they should be perfect. They are the exact same people at the exact same time. But the absolute differences are pretty large, I would say. And so we're exploring some of these potential lab and batch affected probes. That's one of the things we're trying to go forward. There's actually a whole other line of the work that we need to be doing, which is trying other QC pipelines, right? We just picked one. If we did different normalization techniques, if we did different cutoffs, would that make it so that these were far more comparable? You can imagine this being a nice way to test out a pipeline and say, oh, this is the best pipeline we can do for comparisons, for harmonization, and what are the costs and the benefits of doing that. And that's it. That very quickly. Questions? Yeah.
That was great. Would you consider pre-selecting probes on coefficient of variation if you're training a new clock?
So that's a great question. It's actually something that I thought about a lot, less so with this, although I think I would. And so I have kind of two thoughts. One, I've been thinking about this. We have a longitudinal study And I've been thinking like, oh, if you're gonna look at change, why not just look at the probes that we know change? And cut out all the other probes, right? In the same way, you could say, oh, here's our coefficient of variation. We, like this subset, we'll post this list and maybe everyone should stop using these. I like that idea, because then people would cite the paper. I don't like the idea in some ways because I think a lot of our reliability, like when people say, oh, this is a reliable probe, a lot of those are study specific. And I'm still trying to sort out how that is. I thought for sure there was gonna be a subset that are like, these are the bad probes. And actually, if you like read some of the Neaton papers and some of the other papers I follow, they talk about it. So it's a real, this is our subset that is absolutely the reliable ones, and these are the poorly reliable ones. There's two problems with that. One, it goes back to that variance thing. A lot of the probes, you can't calculate a reliability, so they look really, really bad. But they're actually very consistent. It's just that there's no variation. Most of the probes are that way, actually. The other thing is there is that subset of even variable probes that just seem to be slightly different each time. And so you take a subset for HRS and you compare it to another study, there may not be more than like a 5 to 10% overlap in which probes are not reliable. And I think that's what worries me. At the same time, this is the same samples and it is like different labs. So in theory, this might be a little bit stronger. We're gonna put it out. That was a much longer answer. I think the short version is yes. I don't know, did you have any more comments on that?
No, I did a good answer.
Okay.
Okay.
Thanks. Go ahead.
Very interesting talk. We also have lots of examples and stories on batch effect in our lab.
Yeah.
So I have two questions. One of them is related to Oliver's. Did you check if CPG sites that are used in the clock are enriched for more stable
Yeah.
CPG sites according to your data?
Yeah, that's the answer to that is no. I asked our bioinformatician to check that, and he hasn't gotten back, so I was like, oh, I was gonna add that in, but it's a great idea. So that's one of the first things that will come out. It's like, oh,
Ideally, they should be, right?
Yeah, which ones are showing up in here? And which ones, are there any that kind of fall into this subset of problematic ones? Yeah, what was your second question?
And second question, was the position of samples on the plate the same across labs?
Yes.
Okay.
It's identical.
Because what we also see in our lab, that actually position on the plate sometimes also matters.
Yes. Yeah.
And for example, it may be the case that samples in first cone are always on average older than samples on
Yes.
regardless of variability.
Yeah, no, there was a part of us that was tempted to try to mix it up, but I think it is better to keep it exactly the same. So these plates were identical as they were shipped out. I think that was important. We were trying to control as much of all of those sorts of things. So, I sat there and we made our plate manifests identical. Yeah, that's a good question.
Okay, thank you.
Other questions?
Are you planning to keep this blinded the whole time so that we don't end up with this conclusion that if you want your epigenetic age, get it done in Minnesota?
Why don't you just say you wanna know which one was Barat's lab?
Minnesota had the lowest estimate. Does it mean anything? No, but.
I mean, yeah, I don't know. I mean, that is something for all the labs will have to tell us what they would prefer. But I think for our internal group, I mean, you already know which lab is which.
Right, I do.
So I think that's important. I think going beyond, like, I don't know that it's important that the study, that everyone.
Well, what does Tilda think? You want it a secret?
No, I mean, for the paper, I assume you're gonna disclose where the labs were, I think.
I'll describe, yeah, but do you want, anyway, we can
We can talk about that.
A Northern European island.
Oh, no, I think.
Trees.
Revealing which labs were involved is totally fine. I
Yeah.
just don't know like which labs, like, because
Got
none of them
what
are really
numbers?
problematic, but.
I think
I mean,
from a social scientist perspective, it doesn't really matter which lab, because I'm not at the
Right.
same level as you guys. What matters to me is, people are concerned about precision in the social sciences, but even in the physical sciences, there are sources of variation that we don't even think about in the social sciences. So
I think people will pay more attention if they just say, oh, this paper compares this to this, And those people got a 10-year difference.
Yeah.
I think that's the most important thing because it highlights that point that you made at the start that these things are not ready for individual level prediction.
Yeah.
The way they're being sold and marketed. So
They're not even ready for mean comparisons across
Across,
countries.
no, so that's one
It's
thing.
not just individuals.
Yeah, it's
Yeah, the sale of it is a scam. Yeah.
Well,
Yeah.
so I mean, to your point, I mean, I have heard this, people say like, oh, look at our age acceleration is
Exactly.
lower than HRS's,
Yeah.
therefore
Yeah.
we are a
Healthier
healthier.
country. Yeah,
That's
That's
clearly
yeah,
not true.
yeah.
that's the,
Yeah.
I mean, it just makes the case that the number really doesn't matter relatively.
It's just whether or not there
everybody's-
is age acceleration that matters.
I mean,
Well,
I think it's
no,
the
it's how
relative.
much relative
Yeah.
variability, I guess there is.
Yeah.
Which they match very well.
Yes,
Yeah.
the relative scale, yeah, the relative, like, they're highly correlated, but.
Well, I mean, we've done this with many biomarkers.
Yeah.
We get the same idea that there are very good labs everywhere.
Yes.
That their results correlate 0.989, but the numbers are quite different.
Mhm,
Yeah.
And so social scientists tend to think the number is real
mhm.
from anything.
That is true, we do.
And then that it means that that is an exact number in all places and in all
Yes.
times, and that's the problem.
And when you get reviewed for publication, like we used a binary, was there evidence of age acceleration or not? And they were like, you can't tell us how many years? Like the publishers want to know how many years and
Right.
just can't really reliably say that.
There is examples of this going back over years too, where we're looking at cross-country difference between the UK and the US, for example, which are based on HbA1c or something like this, and people are making conclusions that.
Well,
Yeah.
All
But
this
we're
thing
more
about-
diabetic.
Yeah,
than-
exactly, yeah,
Yes,
so
yeah.
that's,
All
but
this
they
like-
understand that they're done in
Yeah.
different labs.
Yeah.
It's using different
Not
assays.
only that, but they're based on the UK, where they dumped the blood in a mailbox and it sat for 3 days and then it got to the lab. They were all wrong. All that thanks and,
All right, well, next presentation is a fairly quick kind of
It's true.
method piece that EunYoung is going to be presenting.
Hi, my name is EunYoung Choi. I'm a postdoc working here. So this is collaborative project that I work on with Eric, Jennifer, and Eileen. And I think this is a nice continuation of conversation on the variation, the source of variation, and we are tapping into another dimension of variation, which is timing. Okay, so I remember that this whole conversation began by several studies that we came across last year that demonstrates that epigenetic age can fluctuate in a single day or by season. And there is another key study that published last year, which really drive the key points to home, which says that epigenetic clocks, because epigenetic clocks reflect the average DNA methylation signal across multiple cell types in a tissue, any variation in this composition, such as by circadian rhythm, can impact the interpretation and estimates of these clocks. So to take a closer look at this individual studies that we found, one study used a blood sample from a healthy 52-year-old male every 3 hours over a 72-hour period to estimate epigenetic age for several epigenetic clocks that we have been using. And what they found out that out of the 17 clocks, 13 showed oscillations. And if you see the graph over here, we see that around the noon, it is oldest, and around the midnight, it is youngest. So I guess we are all oldest now because it's around the noon. And they also find the same patterns using a sample of 31 individuals. So this kind of confirming that what they found is not just a one-off result. It's pretty consistent in terms of their findings. And there's another literature showing the seasonal patterns in DNA methylation, although we couldn't identify studies looking at the seasonal patterns in epigenetic age yet. So this, the seasonal patterns make sense because season is linked to both environmental and behavioral factors such as air composition, daylight exposure and temperature, and other health behaviors such as diet and exercise. So this study, in an Italian randomized study, they took the blood from the 88 healthy male heavy smokers, and they measured the methylation levels in this line 1 region, and they found out that the methylation levels were higher in spring and summer versus fall and winter. So the conclusions that these studies made really got us thinking about the implications for larger population studies. The authors from the studies mentioned that failing accounting for daily oscillation or seasonal oscillation may actually compromise the reproducibility and interpretation of these results because the increased variability that might distort the magnitude or significance of the biological effects or the associations of epigenetic clocks with health outcomes. So the really the overarching research question that we had is, if timing effects matter, how much do they matter for large population studies? And specifically, we have several research questions. One is, do epigenetic clocks vary by time of day or seasonal blood collection in a large population study? And can this variation be attributed to timing effects or other effects that existing in large population study? And the third question is, does this variation affect the association of epigenetic clocks with other health outcomes? And we used the data from the 2016 health and retirement study that have been introduced several times in this meeting. And we used the venous blood, so the DNA methylation sample, which measured the epigenetic clocks among adults aged 56 plus. And this is the basic demographic characteristics of the sample. And they do have the data on the date and the time of blood collection, which allows us the opportunity to examine these research questions. So time of day, just a quick distribution of the time of day in blood collection. We saw that a lot of 1/5 of the sample had their blood collected around noon, and most of them actually got their blood collected from 8 a.m. to 1 p.m.ish, which is, I think it's because this is the working hours. So that's that. And we compared the basic demographic characteristics in terms of their blood collection time. And we found out that younger respondents were more likely to have their blood collected earlier and later in the day. And this makes sense because that's the working hour for younger respondents. But we couldn't find any other demographic differences in sex, race, ethnicity, and education levels. And this is the graph showing the green age differences, and this is not acceleration measure yet. And x-axis shows the time of day, and the y-axis shows the years. And we see a clear diurnal pattern in epigenetic age. So samples who had their blood collected near midday had significantly older epigenetic ages. And if you remember the graph that I showed earlier, this kind of really matches the results from past research, where it shows that around the noon, the epigenetic age estimates kind of get highest. However, when we use the accelerated epigenetic age measure, which is basically regressing age out, we saw that the patterns are flattened. So this pattern really reduced just by controlling for chronological age. And we found same patterns with other epigenetic clocks. This is the results from the whole birth age. And for DNAm pace, there wasn't really much going on from the beginning, but we do see the slight reduced patterns, but essentially, this also matched with the prior study showing that the one of the clocks that didn't see daily oscillation is actually the median pace. So this matches with earlier evidence. And then we went ahead and tested this question of does time of day affect clock's association with other health outcomes? So in this graph shows the regression coefficients of each epigenetic clocks predicting mortality. And in this model, we control for the age, sex, race, ethnicity. And as we are all familiar, higher levels in the epigenetic clocks predicting higher risk of mortality. And then in model 2, we added time of day, and we see that time of day, the additional time of day really does not change appreciably association of epigenetic age with health outcomes. And these patterns were found the same with other health outcomes, including disability, multimorbidity, and cognitive impairment. Okay, and then we also tested the patterns in month of year to kind of figure out the season effects. And to begin with, I wanted to introduce this distribution of blood collection in the health and retirement study that we used. So we can see that it started from April, and then by October, half of the sample had their blood collected. And then by the next March, 90% of the sample had their blood collected. So it pretty much covers all the seasons in the year. And then some people had their blood collected very late until the September of the next year. And again, 1/3 of the sample had their blood collected in summer. And this shows the differences in demographic characteristics of different season. And we see that older female were more likely to have their blood collected in summer than fall, winter. And it's because HRS started in April, and older female are more likely to participate the survey, and that's one of the reasons why I see these patterns in age and sex, but we couldn't find any differences in race, ethnicity, and education levels. So we see some of the seasonal patterns in epigenetic age. Again, it's kind of the samples collected near mid-year, May, June, July, significantly older epigenetic ages. But then we can see that the oldest is not actually the summer, it's May, and it is youngest in March. And when we control for the age, so when we use accelerated epigenetic age measures, those patterns are gone. Same with FH. And for the median pace, again, there wasn't really anything going on from the beginning. And again, the associations of epigenetic clocks with health outcomes were nearly unchanged by addition of month of the year. So our main takeaways first, there are some diurnal and seasonal differences in epigenetic age measures when we don't control for age, but these patterns are mostly derived by chronological age differences that are related to survey response patterns. Timing of blood collection does not appear to have substantial impacts on the association between clocks and health outcomes when we control for basic demographic variables. And that's it.
Thank you. Very interesting data. I'm still trying to digest it. So did I get it correct that the difference during the day that you observe in prediction of the clock, I explained by the fact that blood from older people is collected during the daytime versus blood from younger people is collected during the extremes, right?
Yes.
But how then do you explain the original study where the same person was tracked during 3
I
days?
think that's we can correct me. So my response is that I think that the earlier study looked at the individual variability and what we are doing is the population level. So that can be tied to the why we don't observe those biological patterns that were observed in individual that focus on individual variability.
I think that's the real point, but I would also like to say in one of those studies, there was one individual
Yeah.
followed for 72 hours who was woken up every 3 hours to have a blood draw. I don't know how normal their diurnal pattern was to begin with, and the other one was in 88 Italian heavy smokers. So, and I don't know how much these are like useful as, I mean, the deep phenotyping is useful, but I don't know. This is maybe not the perfect way of calculating these things anyway.
I would be deeply skeptical about these earlier studies that show. I mean,
Responding to reviewers.
Yeah.
Normative aging, they always control for these things. And, oh, you guys are all wrong because you haven't controlled.
I mean, and I would actually, in the takeaway points, I would say there is actually no diurnal or seasonal variation.
Yeah.
Because it's all just age.
Like a bivariate association, but not a.
Exactly.
Right.
Yeah, but like, so Eileen and Eric were working on something and then they were seeing this thing about time of day effects, but like a very strong conclusion about it.
Yeah.
Separately, I was doing the paper on clocks and air pollution, and one of the studies was adjusting for temperature. And we couldn't figure out why, and then we went through their citations to find the paper they cited, which cited a different paper as the rationale, which cited different, they just kept citing themselves, it became a snake eating its own tail. But so we just feel like we need to get ahead of some of these conclusions that are coming out that don't make a lot of sense for the work that we're doing.
It's a beautiful paper. It's great. Yeah, it's really.
Well, it's really clear.
It's
in the
very clear.
Conclusion.
Thank you for doing it.
It's gonna be like,
Well, because now you guys don't have to
Yeah.
do this
gonna
for
be like,
every
why do we
single
care?
analysis.
we don't.
Yeah, it's kind of the null results meeting.
All right, thank you, EunYoung.
Thank you.
All right, here comes another boring methodological doctrine. This actually started out from a more kind of emotionally vivid place. So for years, I just sit there in my office and people show up and say, Cole, I just got my epigenetic age read out. I'm 10 years older than my chronological age. So I said, well, I took the day off of work, I wrote my will, I made a list of people I'm going to mend the fences with before I go. And you can't take this stuff that seriously. Its measurement is not that precise. It varies from day to day. And they just say it's going to be okay. Just go measure your epigenetic age again next week and see what happens. And they never do this, right? But even worse is the undergraduates that show up and they say, Professor Cole, I'm 21 years old, but my epigenetic age is only 17. I feel like I'm falling behind. How do I catch up? I just think, no, don't worry about it. It moves around over time. Just keep, try measuring it again. They never do this. So at some point, after telling people for years and years and years and years that your epigenetic age varies over time, I thought I should actually check and see whether that was true. So the study I'm about to tell you about was this sort of like opportunity to evaluate this in a kind of an unusual format. But I was pretty confident that these things really do vary over time, both from the technical replicate studies that you guys have talked about, but also from what I know about, sort of how the immune system behaves differently as a function of the life that we lead. So, as articulated earlier, the cellular composition of your blood is a huge determinant of what your sort of aggregate methylome profile will be, and that's got to impact the clocks in some way, some clocks more than others, depending upon how they're put together. But the upshot is, at some point we know, there's definitely assay variability, but there's probably sort of, you can think of it as true score variability, true biological variation in whatever molecular characteristics wrapped up in cells constitutes whatever sort of molecular age measure is for any given clock. So we basically set about asking how much does a person's epigenetic age change from day to day? And that's really aimed at answering, you can think of it as the retail consumer questions. If I've got, 5 years, they're like, yeah, 1 year, I'm not that worried about. By 5, they start stressing. By 10, they're literally freaking out. And so we figured, okay, let's do an arbitrary 5-year acceleration. If I've got a 5-year acceleration today and I do another test tomorrow, am I still going to have a 5-year acceleration? Is it going to be a 4-year, a 6-year, a 40-year, that kind of thing? So that's what we were mostly trying to understand. And the way we went about it was by doing some repeated sampling on a single individual. Oh, actually, Em got an earlier version of my talk, or sorry, yeah, Donkey did. Anyway, the way we did this was by retesting a single individual over time, actually 85 times over the course of 329 days. And she was kind enough to record a whole bunch of parameters on the characteristics of her life. So whether she was sick, social conditions, I'll show you guys in more detail the kind of stuff that she looked at. How much sleep she got, how much exercise she got, all this other kind of stuff. She was a UCLA undergraduate, so we also kept track of what's going on in life, like is it spring break or finals week or that kind of thing. So this is, at this point, after everything you've heard, this will not surprise you. So these are just box plots of your epigenetic age for this 22-year-old individual on a bunch of different clocks. So you can see that there's actually quite a lot of diversity of opinion among these clocks about whether she's going to die soon. For that original age clock is actually, to its credit, doing more or less what it should. It's got a sort of a central tendency about her actual chronological age. It's, mushy, plus or minus maybe, depending on whether you want to, what you want to use as your measure of dispersion, standard deviation, interquartile range, that kind of thing. But the sort of, you can think of it as the lobby imprecision is probably about 10 years, 20 years, something like that. So not very tight, but on the other hand, the thing at least is not systematically, if you will, miscalibrated. If you want to feel better about yourself, at least if you're her, go with pheno age. It says you're definitely decelerated by something like 5 to 10 years. In fact, sometimes it says you haven't even been born yet. Negative epigenetic age. I'm not calculating these things. These come from Clock Foundation. So how they, to their credit, they're not censoring bizarre, impossible results, but nonetheless, it gives you some pause about, maybe I should be answering these people not by saying test yourself again tomorrow, but it's like, yeah, just choose another clock because you'll be fine if you're on Hanem or pheno age. By the time you get to Grimm age, it turns out she's a little bit accelerated. Interestingly enough, if you go just to Grimm age 2, which most of you will know is like a tiny little elaboration of the original Grimm age, suddenly she picks up an extra 10 years of life just from the epigenetic report on CRP. I can't remember what else is different in Grimm age. Maybe you guys are.
HbA1c and CRP is a difference.
Yeah. So CRP is probably the sort of major determinant here. This is not an overweight person, so I wouldn't expect it to make as much of a difference as it did. But this is actually an important lesson that was kind of coming out in sort of small form earlier in our discussion, but it's worth really kind of spiking the ball on, and that is. It turns out your Grimm age 2 is correlated with your Grimm age about 0.93, but they're not giving you the same piece of subjective information here, okay? Correlations are not the way to measure calibration or accuracy. Correlations are just basically a giant conspiracy, but conspiracies can do all kinds of crazy things without you knowing it, and that's one of the difficulties. You can have things that are highly inconsistent in terms of central tendency, nonetheless correlate quite well, as in this case. The other thing to appreciate is there's a fair amount of variability in some plots, other plots much tighter. We know that Grimm age is pretty well behaved in terms of its sort of, you can think of it as like random error management compared to the earlier generation clocks. Dunedin pace we'll talk about in a minute, but it generally shares pretty similar properties with Grimm age, although it comes to very different substantive conclusions. So again, if you don't like your Grimm age, go to those pace guys. You'd probably be a lot younger. Everything's going to be great. Standard deviation of these things quite large. It's better for the second generation clocks. but certainly a fair amount of variability. So the upshot is if you are, age accelerated today, there's actually a pretty good chance that you'll be substantially less accelerated, maybe decelerated tomorrow. In other words, the standard deviations are generally larger than the actual average. And I like these averages because when do you get an average of like 85 different reads on a particular individual? Yes, your complaint.
No, not, just the 85 samples, were they all run at the same time?
They will run at the same time on the same chip by the same people. We are 100% on board with everything you're talking
No,
about.
that was my own, but
Yeah,
just
well,
clarify.
you can have more. The bigger issue actually isn't the standard deviation, which you all remember is actually just the average excursion. I mean, when you're trying to communicate to people in the retail general public, they're really concerned about what you could think of as at least, whatever, a 90% confidence interval or something like that. That's when they start like, I get things go wrong, 10 or 20% of the time or whatever. So if you look at the total range these things are off, it's often, decades, literally. So it's to the point where like definitely in the form that we're producing them right now, they are just not useful information, but they're often, quite alarming and in a way that's very effective for revenue generation. Yeah, sure.
Sorry to interrupt. I understood grim age doesn't predict your age directly, it incorporates age into the prediction.
That's correct. So we fed her constant age 22 into that. It also incorporates your sex. So there's.
So that's why it's probably.
That's part of it. They also did a pretty industrious effort to sort of probe screen. So they basically threw out a lot of high variance probes that they believed were predominantly unreliable as the source of variance, as opposed to some kind of like biologically informative version of this kind of stuff. So there's a couple of different innovations that lead to those kind of tighter variances. But nonetheless, I mean, even between grim age and grim age 2, there's still considerable differences of opinion about how accelerated this person is, to the point of one of them, saying she's about 9 years older than, her chronological age. So this is, sort of the prototypical, just correlate everything with everything else. And really the only take home point here is so remember, there's no difference in chronological age here. Okay, so you can take off all of that kind of thing. This is one person with one age. Even if you allowed that person to grow, sort of like whatever, 4/5 of a year older or something like that with a regression equation, still doesn't change any of this kind of stuff. So the basic age acceleration / error, whatever you want to think of these residuals. I mean, we often interpret these residuals as mostly signal, but if you're a psychometrician, you're like, yeah, that's probably mostly garbage, right? So it becomes an empirical question. And so I'll show you a little bit later how you go about actually disentangling those kinds of things. But the upshot is there's very little correlation between these scores in terms of, sort of the acceleration values. This is kind of an easier sort of heat plot coding of this kind of thing. You can see first generation not correlating very well at all with later generation clocks. By the time you start to get to the 1.5 to 2 generation, there's a little bit more coherence. You can see the kind of the evolution of thinking and reliability there as you move from Pinot age to grim age, and then a sort of you can think of it as almost independent overlap with Dunedin pace. But certainly grim age and grim age 2 correlate very highly, but again, remember producing very different implications for actually whether she's going to die soon. So the big question I had is essentially, is this stuff all random crap noise, or is there actually some kind of systematic variation in the biological her that is also driving this variability? So remember, most of the stuff we've seen so far has been the same sample assayed twice. That's a great way to capture assay variability. But very rarely do we have the same person assayed multiple times to get a sense of how much variation in their physiological age is taking place. And so that's what I'm going to actually spend the remainder of the talk showing you. So this is one, sort of appealing clock. This is grim age 2 acceleration in her particular case. This is going to turn out to be the most successful of the clocks in terms of its, sort of net bet. This is one where actually only less than 10% of the time do you get a qualitative reversal in the clock's call about whether she's accelerated or decelerated. So it's making a pretty extreme bet that she's about You know, she starts 5 years older than her chronological age, ends 10 years older than her chronological age. This is not impossible for a UCLA undergraduate. Their lives are tough. She's getting ready to go to medical school. That was a very unpleasant summer studying for the MCAT, that kind of stuff. So it's not totally implausible, but actually the bigger thing here is just the amount of jump up and down. And so most of you are not used to looking probably at time series data, but if you are used to it, you'll go, that's a pretty bursty sequence there. It's not the case that you have a homogeneous random spray across this range. There are periods of relative quiescence and then periods of kind of explosive variability. So to help kind of graphically show that, I'm going to put up Levine residuals, which are basically the absolute value of the residuals from the regression line. And you can see there's these little kind of volcanic eruptions, periods where suddenly we have a lot of relatively extreme residuals, a little bit more variability. Something is definitely going on there. So and yet at the same time, those don't persist indefinitely, like they resolve and kind of regress back to some mean level of sampling variability over time. Okay, so we know what might be causing these little eruptions. Well, again, conveniently enough, she recorded information about a lot of things going on in her life. And one of, we can kind of take this stuff, feed it to the appropriate analytics, and what will pop out is insights like this one. So one example of a parameter that she was keeping track of was just, do you feel sick? Do you have a cold, a flu, food poisoning, all that other kind of stuff? So you can see those little green tracks down there. Those are her ratings. They're, low when she's feeling well, one click up if she's feeling not great, and another click up if she's just like, dead in bed or something like that. So one thing you can see, and it'll be easier if I put little boxes around it, is that during periods of illness, in retrospect, unsurprisingly, these are basically, leukocytes that are driving the methylome in these kinds of whole blood samples. There's this big spike up in her Grimm age acceleration. This actually doesn't start right away when you get sick. It has a lag that's probably related to cell production as the immune system starts to clone like crazy. This is probably not coming out of the monocytes. They probably do affect other clocks, but for this particular clock, it's probably picking up the burst of, sort of T cell and B cell proliferation that takes place after the antigen specific immune system really gets its flywheel revved up. Yeah.
Sorry to interrupt you, but I noticed that you're talking about I'm assuming a biological female who's fertile, and yet there's no mention of her menstrual cycle in the.
Well, you're right. We
Or birth control.
Yeah. So we have to be very careful about what we ask people about. So the way we manage that is we let people generate their own vector of things that they're going to report on. So it's actually essentially a student self-research project. So in this particular case, we can't really address that, but it's an excellent point.
Because that could be also when feeling sick could be her way of talking about cramps and all that stuff.
That's true in principle. You could, assuming that she's on a reasonably regular cycle, you could actually sort of take that kind of stuff out. We do have other undergraduates who have that parameter in their stuff. We just don't have methylation data analyzing. So it's a potential explanation, but you'll see as we look at other periods of illness that are, sort of noticeably less than a month duration, that, you can clearly still separate out something. You can think of it as like a monthly cycle, idealized, imputed, or something like that. So that may well be influencing things here, but this kind of illness burst seems to be at least a little bit above and beyond that.
Okay, thanks.
So you can see a couple other instances where she got sick. And there's again this kind of big lagging temporal variability burst. Usually lags by about 4 actual measurement occasions. She's doing measurements 2 to 3 days apart because you gotta heal all those holes in your arm somehow. So they give her credit. But yeah, interestingly enough, the big variation, at least in Grimm age 2 acceleration, seems to lag the onset of illness symptoms by maybe 4 to maybe 10 or 12 days, depending on the circumstance. All right, let's look at another measure of age acceleration. This is Dunedin pace. One of the things you'll realize is that dashed line is right in the middle. Okay, so Dunedin pace does not agree with either Grimm age or Grimm age 2 about whether this person's going to die soon. It thinks she's just like oscillating right around 1. She's doing fine. Maybe she, on average, I believe she's about 2%. She's like at 0.98 on Dunedin pace. And I think the standard deviation is about 14.14 here or something. Oh, I was amazed at how close it was to numbers that Colter was showing. So it does, does seem to be kind of par for the course in terms of the absolute magnitude of variability. Again, you can see, I'll put the little kind of variance volcano plot up there. You can see there are periods of variance eruption, but they're not taking place on Dunedin Pace at the same place that they were taking place on Grim Age. So I'll show you the little purple boxes I showed you, areas of illness where Grim Age was starting to flop all over the place. And Dunedin Pace actually doesn't seem, unusually variable there. Sometimes it gets a little crazy afterwards. Sometimes it's a little crazy beforehand. We can't really figure out exactly why that is, but you can see that, so the magnitude of these variations is quite substantial. But in any event, Dunedin Pace doesn't think that she's markedly accelerated or decelerated, but it does generate very unstable opinions about that during certain periods. So no association with illness symptoms. What we do is we take that big vector of life parameters and we throw the usual machine learning stuff at it and ask, what among these life parameters that she recorded actually ends up collectively predicting these age acceleration values. So in this particular case, for Dunedin Pace, the things that turn out to matter for her are periods of academic stress, periods of good mood, and periods of UCLA finals. Strangely enough, when academic stress and UCLA finals come on, her age acceleration goes down. So like stay stressed the rest of your life and you'll live forever, but you'll be suffering the whole time. Mood makes more sense in the sense that, sort of the higher her positive mood essentially is, the lower her Dunedin Pace score. You can do the same thing for something like Grim Age. And what will pop out is actually, it turns out a very different set of predictors. So for Grim Age, what's going on is first there's a secular trend. Like I said, she aged, epigenetically about 5 years over this kind of approximately 1 year period. So there's definitely a secular trend up. She was doing this stuff during the era where COVID was still afoot. And so the university kicked her out of her dorm when she got sick, sent her home to her family, associated with reduction in her Grim Age. That sounds good to me. And then during UCLA breaks, she also had a pretty substantial reduction in Grim Age. So the take home point is that different life circumstances seem to matter for different types of clocks. Like the clocks, because they're built usually with some particular kind of, almost like conceptual strategy in mind. Those conceptual strategies are different. They're going to be picking up different biology streams and probably be affected by different exogenous circumstances as well. So you've survived. I'm almost done. Don't worry. The take home point is that most of these epigenetic clocks have sampling variability from a day-to-day point of view that is substantially greater than their average degree of acceleration. So that sampling variability is to some extent due to measurement variability, but at least as much and sometimes more, depending on the clock, due to true biological variability in how light is affecting essentially the white blood cell pool that ends up becoming your methylome for this particular sampling procedure. So most, as far as I can tell so far, all epigenetic clocks show substantial daily sampling variability, with that variability often greater than the average acceleration in years terms. So epigenetic age is highly variable over time within an individual. Like I said, the range often exceeding the average value. An accelerated individual, depending on the clock, could very easily be decelerated tomorrow. These variations are not totally random. They involve some temporally correlated bursts of variability that seem to be influenced by illness, social conditions, stress, mood, that kind of thing. Those bursts of temporal variability tend to be transient within the course of maybe a weekish or something like that. They generally regress to some mean level of variability after a few days. And we can do, traditional variance components analysis. We invested in replicate measurements of a couple of these things, so it can give you a reasonably quantitative sense for each clock. For some clocks, measurement variability is about 30% of this, and presumably the remaining, whatever, 60 to 70% of variability is due to true score variability in your biology of epigenetic age. In others, the ratio is about inverted. So it's the span is about 1/3 measurement variability, 2/3 measurement variability, with the complement being this kind of systematic biology. Clocks do differ in variability across them, as well as in their verdict about acceleration versus deceleration. Second and third generation clocks show the lowest retest variability, but even among them, standard deviations are still high, and external influences do affect them, but different influences affect different clocks. So given the magnitude of variability here, we would say conclusions about individual age acceleration are quantitatively imprecise, to the point of being qualitatively unstable. I mean, in most cases, you could be just as easily decelerated tomorrow as you are accelerated today. So, probably the biggest take home point is these tools, and they're great tools, don't get me wrong, these things, presumably some of these clocks are right about something in terms of your health risk. And actually, that's how I don't think of them as clocks, because I can't see where their long-term predictive value is actually coming from. I think of them as sort of multi-component aggregated health indicators, much like an allostatic load measure registered through a particular kind of, methylation lens, and you can do this at lots of different levels as well. So at the same time, as we're now trying to sell these things to individuals, or even trying to make individual predictions, they're really only trustworthy, it seems, at some kind of aggregate trend level. They're not suitable for an individual, sort of action like changing your behavior or your health or your medications or your senolytic consumption or whatever, unless they're repeated. So if you don't like your don't worry, just try it tomorrow. That was probably the right answer. I'm not sure if I have the graphics. I think I ended up with a slightly older version of my talk. So I'll actually, in the interest of time, kind of skip the discussion of how we can actually chop up the total variability into measurement noise and true biological variability that most of you were used to thinking about. Analysis of variance decomposition kind of get this pretty quickly. And with that, let me thank you for your attention. Or that's how we do that. The last thing I did was I basically calculated how many times you would have to sample in order to get a 90% consistent call about whether you're accelerated or decelerated. And that unsurprisingly depends on how extreme the bet was in the first place. So Grim Age 2, you'd really only have to sample maybe 2 or 3 times before you're about 90% consistent in claiming that she was accelerated. For the others, there's, Horvath 1 and Dunedin Pace were more or less saying she's not really accelerated at all, so I don't really penalize them. But for some of the other clocks, it was really like you needed, anywhere from 8 to 24 different samples before you actually settled down to about a 90% reliable call without acceleration. So, sorry for talking so fast and getting you guys with so much stuff, but at least we get it to a bunch of years.
Considering this variability and then also population variability, have you done any estimates, any back of the envelope calculations about sample size and power of detection? Like what kind of sample size are we talking about in order to see a difference? And how, I remember from the first talk today, she was saying that it's a relatively stable measure and you don't need a large sample size, but this makes me go.
I haven't done, sort of a power factor calculation for, let's say, study total sample size, but I think that should be linearly dependent upon the standard error. So if that's the case, then in a newer version of my talk that is kind of like missing or has a graph that's not present here, you can actually see for different clocks, the answer is quite different in terms of how much you'd have to adjust sample size. And you can use these kinds of sampling variability estimates at the individual standard deviation level. Basically, you just end up multiplying or dividing by the square root of n, and you ramp up the n's to the point where your standard error gets to wherever you need it to be for whatever your substantive purpose is. And that standard deviation or standard error starts off with actually dramatically different scale across different clocks. So a lot of it's going to depend on which clocks you want to use in the first place.
But so say, for example, for HRS sample, is that even like what kind of effect size can detect in the sample, say a couple thousand?
The answer would depend on which clock you want to look at, and also, I haven't done the back calculation in any generic sense that I could kind of, give you a range of how much it is, but the good thing about HRS is it's effectively asymptotic in terms of, sample size. They're at the point where, like, if you doubled the sample, your increment to statistical power would be pretty negligible. So I think the bigger question is, given where we're at, like, what could you do? Even if you calculated, like, we're stuck with this all the time in clinical trials. We're like, yeah, 2,500 people for my trial. Awesome. I've got a budget for 40. So that's what we're gonna do. So my sense is, you could do that theoretically to assuage a very, sort of like mathy analytic reviewer, but there's actually not practically that much you could do about it because I don't know, maybe you've seen something different, but most of the time I was hanging out with HRS, they were not saying, yeah, let's just expand our sample size by tenfold.
So I was wondering, in practice then, if people are doing additional tests following themselves, would it be adding sort of the variability Colter was talking about to what you're observing? Is someone doing it every 3 months from these companies or something? So would it be even more?
I'm not sure, given the rate of bounce I saw, I'm not sure that 3 months even makes sense. It might make sense if there's a big secular trend. So be aware that I'm talking about a UCLA undergraduate. Those are among the healthiest creatures in the world. There's very unlikely to be some, sort of nascent chronic illness thing that, is kind of rearing up out of the muck of her life. So I think we still don't know sort of what, we don't know the effect size that is sort of clinically or biologically important to detect. Once we knew something like that, we could probably do the calculation that you advised, like, would it be okay? Like, most of the chronic diseases that we worry about are slow onset, long-term construction processes. So 3 months actually might be fine.
Well, I guess I'm wondering if it's even worse than you were suggesting.
That's always
people-
my thinking.
Yeah.
There's a
doing their
lab
own-
effect and
There's
there's
a
a
lab
batch
effect,
effect.
right.
There's a batch effect, there's a lab effect.
And so not
on
only
top
are
of-
these like
yeah.
tests like really deceiving and stealing people's money, but I guess for longitudinal studies too, I'm wondering, do you recommend,
I do recommend
follow-up?
continuing to use these things because I have never seen any evidence so far that makes me feel like they're biased. I think they're noisy, but I haven't seen, even that, like, it's not like I believe that they're unbiased. I think we still just don't know what these individual clocks are really doing. We know.
Right, but like, does the outcome change over time across surveys?
It will probably change over time, almost certainly. How well we can resolve that, given the signal to noise ratio, is a question. But, the thing that population health always has on its side is this massive overpower, right? And so sometimes it's easier to just do more sampling than it is to try and tweak on your biological metrics to try and make their variability lower. And, the central limit is like God's gift to research. You can solve so many problems by just sample, and pray to God that this variability is not correlated with what you're doing. You've got an unbiased measure. So I retain a fair amount of enthusiasm for the clocks, at least relative to what I started with, on sort of their utility for, nomothetic population health research, where I think we're all converging is like, these are not ready for, clinical use or direct to consumer diagnosis. And in fact, they're probably great ways to scare people and alarm them.
Is this published? Is this published yet? Okay, so it hasn't been reacted to by these companies.
Well, actually, that's why it won't be published.
Oh, it won't be published?
Well, that's part of it. The other part of it is it comes from undergraduate research projects where, we essentially have to, if they don't want us to, publish their data, if they want us to, they'll give us like different levels of sharing. And so to do something like this, first, we would have to do more undergraduates than we already did. So that was about a $20,000 project there. So we'd have to probably do this at, sort of bigger scale for it to really be
You
quote,
pre-print it or
generalized.
something like OSF or something
You
for people
can't,
like
you don't think you can put it out? You don't have permission from?
We don't have permission. Yeah, so we have permission to share at a certain level of, sort of specificity, which is consistent with what I presented today. But we have to honor their requests.
Wait, but you,
They're stressed and shy about.
you have a permission to share the level you presented today. But why, so you don't think you could publish it?
Probably not.
You pre-print it so people can cite it?
We can,
we put it on the
probably not
website?
even do that, but we can put, a clip of this talk on the website, we can put the PowerPoint on the website.
Put you out of business.
Okay.
I thought that was brilliant. I really, really enjoyed that, right? Yeah, so I'm playing devil's advocate. So I don't believe in the second, right, around about the use of these in individual level precision medicine yet or anything. But I'm thinking of what the likes of True Diagnostic would say, and it's a 22-year-old female college graduate said very healthy. These clocks are designed for utility in older cohorts. They were trained in older cohorts and so on, so we wouldn't expect them to work so well. So I'm just wondering if you do the exact same thing with someone who's like, let's say you take an individual, and you have the budget to this, an individual who's 50, 60, 70 years of age, and then you do the exact same assessment with them, would you expect the variability be different? Because what I'm thinking when I was looking at that, and I saw those periods and ups and down, I've had this when I was in college, we had weekends where you were heavy on the booze, you might have been a little bit more stressed with exams and so on. As I've got into even as I approach my 50s, now all of a sudden I don't do those things so much anymore, right? So that's
Yeah.
well, up until last night.
Want to respond because like UCLA undergraduates do not suffer from excessive drinking anymore. So it's really a profound change in the psychology of undergraduates. There's, for the people I interact with, there's very, very little partying. These people study like crazy.
You need to take a
That
serious
was trained
university
on a
sample because we
Yeah,
have
they
a
did.
bad
They were selling their
record.
phones, so it was 45s.
But your original question,
That 20-year span.
your question is completely respectable.
Yeah.
So the question is like, is there a kind of a true score physiological phenomena to discover in this person? Or are we just picking up sort of, as we move through life, life moves through us kind of stuff, right? So that may well be true. What I can say is just from life moving through a very healthy body, you can get a fairly huge amount of variability. Second, there does seem to be some kind of systematic effects here. So it might well be useful to use this kind of stuff to just understand how, normal human organisms or anything else live and how, then we, in other words, it's not the case that the epigenetic clock is only useful for disease or mortality prediction. You could decide to use it as like a biomarker of interest that has, vaguely plausible implications for long-term health and well-being. That said, I would generally agree with your critique. If the application is to predict disease and mortality, trying to do that in 22-year-olds is kind of a fool's errand. If like again, there's been lots of discussion about like, well, then why are we selling this stuff to these people on Valentine's Day, right? I mean, that part doesn't doesn't follow exactly from that. So I think there's room to ask a question about that, but on some level, you could actually say, well, you can still learn stuff about yourself and how your physiology responds, even as a young, healthy individual. But I generally respect the argument because I agree there's probably not a big phenomena of the sort that we originally looked to these things to do in some kind of, sort of crystal ball way, like find who's failing biologically without deniers. Yeah.
So this was a very interesting talk, Steve. I really enjoyed it. I'm trying to think, like for all other biomarkers that we use in clinical biology, we calculate this index of individuality, we do exactly what you did for DNA methylation. And then we compare, between person variability to within person variability. We create an index, and that forms the basis for deciding whether a biomarker is good for population studies or not. We've never done it for DNA methylation because I assumed it is terribly stable because it's DNA.
No, it's actually just the opposite. So that you're the only person in the room who shouldn't have made that mistake, right? And you're the only one that runs the hematology analyzer. You just got done telling us about all those T cells and B cells. You know their populations go all over the place when you get sick. You also know those guys here, the immune system, sorry, not the immune system, the neuroendocrine system, the nervous system, growth factors, all kinds of crazy stuff changes the relative distribution of these cells, even acute stress, like within 2 minutes, I can quadruple the number of NK cells in there, right? So any methylation signature that tracks with that kind of stuff should be this volatile.
So I guess that's the point I was getting at. Is this variability really related just to the distribution of the cell distribution? Or is it truly like a DNA methylation difference?
Both of the above. So I've spent, remember, I started off as even more sickly immune person than you by a lot. And so, I spent years going this is the quantitative fraction that belongs to this particular cell type, that particular cell type. I haven't done that so much with methylation per se, but we certainly did that for years with RNA, and that's how we ended up with the tricks we ended up with, which were largely designed to sort of quickly dispatch cell type effects by contrasting things that were taking place in the same cell type, but reading out different signaling pathways, basically, so that, if both of them went up and down, you were implicitly controlling for cell type abundance. So definitely, there is a significant contribution from that. But we have never seen enough information in the kind of flow cytometry that you do so well to say, how is it reading out smoking so well? We've got nothing in the white blood cell enumeration pool that is as sensitive to smoking as, for instance, this DNA methylation signature. So there's got to be some of what we're all imagining, which is this slow sort of tonic readout of some long-term exposure to cigarette smoke leading to some long-term propensity for epigenetic marking in a particular type of cell. Incidentally, which the cells you're seeing today, they weren't even alive 5 days ago, right? Myeloid cells are probably the big drivers of this kind of thing, their lifespan. So there's no way that the bad day you had in childhood has left a monocyte marked and it's just kind of hanging around, right? This is essentially a regenerated stochastic feedback system that's continually generating new myeloid cells every single day at the billions or trillions of scale, and regenerating this particular marking idiosyncrasy correlated with smoking.
Also, that study that EunYoung talked to you about, the within day, hour to hour fluctuation, that was up to 5 years of difference. They attributed it to cell type fluctuations throughout the day.
Yeah, and this actually was what, the first generation clocks, they surfed on this general trend towards increased myelopoiesis in general, and especially increased neutrophils to a lesser extent, things like that. And eventually, Horvath got smart and started going after naive versus sort of senescent T cell and B cell signatures. But originally, it was almost all this myeloid cell. And, I still remember you talking about it. You're like, yeah, this is mostly just a proxy for the number of neutrophils you've got.